I would like to welcome Campbell Ingram, the Executive Officer of the Sacramento-San Joaquin Delta Conservancy, to provide some welcoming words. Thanks, Campbell.
Thank you, Angie. Well, good morning, everyone. I'm Campbell Ingram with the Delta Conservancy. It's my honor to welcome you all to the sixth already off my notes, sixth biannual Delta Invasive Species Symposium. Thank you all for taking the time to be here today. I mean, we're all really here because we love the Delta. We understand the significance of the Delta as one of the largest estuaries on the west coast of the Americas. Of course, why are you approaching? Of course, the hub and a major component of California's water supply, but also just a really, truly wonderful place with vibrant communities, incredible agriculture, and an ecosystem, although a challenged ecosystem, but just an amazing ecosystem that supports over 700 different species. And so we know this place, we love this place, it's very special. And we also know, of course, that invasive species just really fundamentally challenge every aspect of the Delta. And that, in a rapidly changing climate, there's ever increasing opportunities for and impacts of invasive species. So really that's, the importance of why we are here today. Really coming together to build the relationships and get updates on the latest science, coordinate our activities to achieve greater impact, and just general information exchange is just so important in this environment today. I think we all recognize that the current federal administration and getting ready to go into our fourth really challenging budget cycle for the state, there's not a lot of money that's going to be raining down on this effort. So really, this is kind of where it happens. This is where we are coming together, building the relationships and getting our shared understanding. And given the climate and where we are, this is really our best defense and in working together and really where it happens. So the Delta Conservancy is very glad to be able to support the DISC team and help coordinate and organize that team since its inception in 2013. And we're also very proud to be able to help fund Nutria, or not just Nutria, but invasive species eradication in the Delta, most notably our Nutria eradication efforts that we funded through Department of Fish and Wildlife. And Jessica Voller is going to be here later today to talk a little bit more about that project. So before I turn it over, I just want to do a real shout out to the organization team. I mean, these people are really volunteers. They do this because they're passionate about this. Sort of extra duties as not even as a sign, but recognizing just the importance and the significance of this issue and the lack of any other place where this coordination is happening. They've stepped up to make this happen. And so I want to recognize Elizabeth Bersati from the Stewardship Council, Gina Darren from the Department of Fish and Wildlife, Tomas Bush from the Department of Fish and Wildlife. I'm sorry, I said Gina, Department of Water Resources, Tomas, Department of Fish and Wildlife. And of course, Angie Shakya and Rachel Wigington from our staff. So thank them. Throughout the course of the day, it takes a lot of effort to organize something like this, and it's get you all here and do the work that we're going to do today. So really appreciate their efforts. And lastly, want to again thank the Freshwater Trust and Westerfeld Ecological Services for providing our refreshments. And thank you all again for attending and for the presenters, participants, and the attendees. It's great to have you here. So with that, I'm going to welcome Madeline Drake, who's the Assistant Secretary for Biodiversity with the Natural Resources Agency.
Thank you, Campbell, and good morning, everybody. It's a pleasure to be here, and it's great to see so many folks coming together for such a really important topic like invasive species in the Delta. As Campbell said, I'm Madeline Drake. I'm the Assistant Secretary for Biodiversity and Habitat here at the California Natural Resources Agency. And this morning, I'm just going to talk a little bit about how addressing invasive species fits into our broader biodiversity and climate goals for the state. So just a little bit of grounding and headlines that I think everyone in this room is all too familiar with. Invasive species are something that impacts everybody from local communities all the way up. So these 4 article headlines here, I pulled from the local news here in California. So not only are folks like ourselves seeing headlines like this, but people who are like my mom, who works at an elementary school, or also have access to these headlines. And they're affecting many, many aspects of life for Californians. So things like invasive insects like mosquitoes, public health, as you all are too familiar with, Nutria impacting our wetlands, invasive plants making our ecosystems more vulnerable to wildfire, and another one all too familiar with, things like golden mussel, who is really impacting our waterways and livelihoods. And so, you know, these are, again, local headlines, but it's really a global issue. And so this is something that's recognized by the United Nations. So as part of the Global Biodiversity Framework, there are 28 different targets for sort of how countries from all over the world can come together to meet our biodiversity and climate goals. And one of those 28 topics is really focused on invasive species, so target 6. And so I'm not going to read this whole sort of description of what target 6 is here, but importantly, it's about how can we come together, all the parties to this agreement, which is nearly every country in the world, to address invasive species, because it is a global and a local issue. So in California, in October of 2020, Governor Gavin Newsom signed Executive Order in 8220, and really broadly, this was to expand nature-based solutions to achieve California's climate and biodiversity goals. And I want to note the word expand there, and that's because there's been a lot of great work happening beyond and way before 2020, right? But what these are really meant to do is sort of take those global frameworks and how we're going to make those a strategy here in California. So they're kind of aligning with those global frameworks and putting metrics into how we're going to achieve those here in California. So out of this executive order came 2 key strategies. One of them is our Natural and Working Lands Climate Smart Strategy, and the other is our Pathways to 30 by 30 strategy. And both of these include invasive species and things that we need to do to manage for biodiversity and climate goals for the state. I'm going to focus a little bit more today, though, on our Pathways to 30 by 30 strategy, which is the commitment to conserve 30% of our lands and coastal waters by 2030. But I just want you all to know that invasive species is included in both of these. So after 18 months of an extensive public outreach and engagement process from when that executive order was signed, the state released our Pathways to 30 by 30 strategy in April of 2022. And so there are 3 key objectives, or what we're really trying to achieve when we're thinking about 30 by 30. That's to protect and restore biodiversity, to expand access to nature, and to mitigate and build resilience to climate change. We also have 3 core commitments, which is to advance justice, equity, diversity, and inclusion, to strengthen our tribal partnerships, and to protect our economic prosperity, clean energy resources, and food supply. And how does this relate to that global biodiversity framework? Well, in California, 30 by 30, we're using it as our umbrella for a lot of the targets, including invasive species in that global framework. And that's because it's so catchy, right? It's really easy to say 30 by 30. It's really easy to put out a sticker with 30 by 30, and it has a clear metric in the title, right? Like we can all rally and get behind a goal of 30 by 30. But there's so much more to what that goal actually means, and I'm going to dive into a little bit today, but definitely encourage you to learn more. So what do we mean by 30 by 30 conservation area? One of the things that was really important when putting together our strategy is, well, how are we going to define that metric? So after, again, extensive public outreach, we came up with the definition of land and coastal water areas that are durably protected and managed to sustain functional ecosystems, both intact and restored, and the diversity of life that they support. So 2 key things to that definition. It's durably protected, it can't be sold tomorrow and developed, and then it has a management plan for biodiversity, really important for how we're going to address things like invasive species. So where are we at today? Well, as of July of 2025, the state has conserved 26.2% of our lands and 21.9% of our coastal waters. That's amazing work. And a lot of this was done way before the strategy came out. We are really proud, though, that since the strategy came out in 2022, thanks to the collective work of many in this room and beyond, we've added 2.5 million acres of conservation areas to this goal. And that's a really important number because that means that we're on track. But that doesn't mean that it's going to be easy. We still have 4 million acres of land left and about 280,000 acres of coastal waters. So our strategy lays out 10 different pathways for how we think we're going to achieve this goal, and I'm not going to go into all 10 of these. I really just want to focus in on 3 that kind of get to the heart of things like invasive species and how we're going to sort of need to address those to really meet our goals. So in pathway 6 is to expand and accelerate environmental restoration and stewardship. So why is this important to us? Well, 30 by 30 is more than just the acres. Yes, it's a catchy headline, but if we're actually going to meet those biodiversity and climate goals, then we need to be managing these lands for things like invasive species. We also know that we need to be increasing pace and scale of what we're doing because, we have these biodiversity and climate crises on our hands. So how do we really meet the moment and get things done on the ground more quickly? So some challenges that we're facing in this area are ongoing funding. Again, I'm sure you all are all too familiar with this. It's really hard for the state to fund ongoing funding, right? Like ongoing management of lands to get that sort of allocation every year. So how can we do a better job of thinking about how we can sort of get those regular allocations so that we can manage for things like invasive species long term? And then also thinking about ways of how we manage those like really large projects that need multiple funding sources or the really small projects where a state agency might be like, oh my gosh, the contracting is too much for such a small amount. So how can we look to better understand ways that we can sort of work together as agencies to fund those really large projects or do things like Brock grants so that we can fund those small, sort of really effective, but maybe a less amount of money project. Pathway number 7 is to strengthen our coordination among governments. So nature doesn't conform to our boundaries, as we all know. So how can we really work together to coordinate our resources and really get a landscape response that we need to these issues? So some of those challenges we're always working on, agreeing to those shared goals, bringing our missions together to meet leadership changes, as we know all too well with our federal government and changing priorities. What does that then impact us and how do we continue to keep our work moving forward? And then enough resources to go around, right? There's a limited number of resources. So how do we make sure there's enough to get the work done? And then lastly, the pathway I want to focus on is evaluating our outcomes and adaptively managing. So we need to learn and grow. That we didn't put out a strategy and then say, that's it, it's perfect. We're never going to go back again and evaluate, right? Like it's never going to work. There's always going to be emerging issues. So how do we learn and grow as we go? And how do we utilize the opportunities of our partners who are coming together on these important goals? So we have things like the California Biodiversity Network who are coming together on a stewardship roundtable. We need to better incorporate invasive species into some of our mapping tools. And we also have a new opportunity. So just in the fall, Governor Newsom signed AB 900, and that's a bill that's going to require the Natural Resources Agency to do a more thorough analysis of stewardship in the context of 30 by 30. So there will be an opportunity for everyone to be involved and share how we can do a better job together in stewarding these lands. So we do have mapping tools available. Again, they don't include invasive species, but they include ways to find opportunities for conservation, for biodiversity, access, and climate goals. And you also can track and see how we're doing in meeting our target. So when we're advancing 30 by 30, we really want to think about funding. We're really grateful for the climate bonds and having that pot of money to implement resources. But again, that's not going to solve all of our problems. So thinking through how we can get projects funded. We have informational resources, facilitating connections, improving our data and mapping, and then continued engagement. It didn't just stop after that 18 months, right? We have webinars, we go to local meetings, we have a newsletter, we have a blog, we have our annual progress report that's actually required by the legislature. And we also have a big summit every year, which we invite all of our partners to come together so we can rally together and really figure out how we're going to solve these needs together. And then thinking more specifically about the Delta, well, one myth I want to bust really quick is freshwater does count. It is included on the land side, and that's because the mapping, we want to kind of simplify it for a large audience. And also it can be hard to map ephemeral areas. So rest assured, freshwater does count. It's just included in the land's percentage. So we really want to uplift the regional plans and strategies that are already out there to make sure that they're getting implemented and that your great work is really sort of, we want to be the wind in your sails. We want to find opportunities for new acquisitions and easements. We want to strengthen management for biodiversity plans. And then we also want to utilize the climate bond efficiently and effectively to get work done. So we hope that you all will continue to engage with us and provide feedback as we sort of go into this new stewardship area and join us in achieving our 30 by 30 goal. So thank you again for all the work you all are doing to help advance our climate and biodiversity goals and for coming together on such an important topic today. Again, here is my email and feel free to reach out if you have any follow-up questions. Thank you so much for having me here today.
Thanks, Madeline. All right, so I've already been up here, but to more formally introduce myself, I'm Angie Shakia. I'm an environmental scientist at the Sacramento-San Joaquin Delta Conservancy and one of the DISC team co-leads. So today we're all here for the 6th Biennial Delta Invasive Species Symposium, an event that's been hosted and organized by the DISC team, or Delta Interagency Invasive Species Coordination Team. And that we short to DISC. So you'll be hearing both on and off today. Okay, so the DISC team was formed in 2013 to foster both communication and collaboration among groups that detect, prevent, and manage invasive species and restore invaded habitats in the Sacramento-San Joaquin Delta. And the group is co-led currently by staff from 4 state agencies, the Sacramento-San Joaquin Delta Conservancy, Department of Water Resources, Delta Stewardship Council, and CDFW. Members of the greater DISC team, we have a handful, probably up to upwards of 50 other folks on the DISC team that come from various federal, state, local agencies, nonprofit groups, and academia. So really wide ranging, and the team aims to achieve this goal here through a few different avenues. So firstly, we focus on the exchange of information. The team convenes quarterly, and at our meetings, members provide updates on their work. So the team gets together to problem solve anything that's going on, and then team members can then take back what they've learned through conversations at our meetings back to their teams and colleagues to further problem solve and just communicate information in that respect. Additionally, at these meetings, we'll have guest speakers that come and speak on a variety of topics related to invasive species. These highlight the importance of strengthening the interface of science, policy, and management for invasive species in the Delta. And then the last point here related to our work on the exchange of information is just that the DISC team organizes events such as this one, the Biennial Symposium, which this year focuses on modeling and management of invasive species. But in the past, some of the themes that have been covered are things like connectivity, EDRR, so early detection and rapid response, remote sensing, to just name a few. So these will continue to happen every other year, and we look forward to having you all next time. All right, another sort of way the DISC team reaches its goal is through identifying management needs and highlighting where research and science could help address these needs. So previously, the team developed a research needs table, which informed the Delta Science Action Agenda, and that was put together by the Delta Stewardship Council. But more recently, and this is sort of hot off the press here, several members of the DISC team have convened to work and create a quick start guide of sorts geared towards land managers, and compiled a list of resources related to managing aquatic invasive aquatic vegetation, excuse me. So if this is of interest to you, we have a QR code here where you can scan, and that'll take you to the webpage with the information, or you're welcome to share it with those that you think would be interested. I see thumbs up, so I'll pause for a second while you scan. And then finally, to build off the last point, the DISC team works to leverage and bring funding to projects that address these identified research and management needs. So here in the photo is an example of a project, the restoration site at Black Block, where there's ongoing efforts to control invasive Phragmites. Okay, so that's a little bit about the DISC team. If you're interested in joining the DISC team, you think that you'd be a good fit for us and want to learn more, you can come find me, or any of the co-leads will be around today. There's also more information on the Delta Conservancy website. And if you Google DISC team Delta, it's one of the first things that pops up. So it should be easy to find. So we're really excited to have you all here today. We have a lineup of exciting talks for you. Here's a little snapshot of our agenda today. First, after our next talk, we're going to jump into a session that's focused on the various ways that we can think about managing invasive species. We'll then hear some updates on the golden mussel, highlighting its impacts and methods of prevention and control. We'll have lunch, come back for lightning talks. The final session of the day is going to cover some of the new tools being used to manage invasives, and we'll wrap up for the day. So my time's up. I'm going to wrap up and hand it over to our last speaker of our opening session, Lisa Marie Wyndham-Myers, Delta Lead Scientist.
Good morning. Thank you again to the organizers for thinking of me to add to the opening remarks with Madeline and Angie and Campbell. My name is Lisa Marie Wyndham-Myers. I'm the current Delta Lead Scientist. I'm on loan from the USGS to the Delta Stewardship Council. And I'm a biogeochemist, and I work on landscape scale and small scales at the land-water interfaces. And I actually got my start in grad school with invasive species. So Phragmites was my focus. And the great thing about an invasive species for studying ecosystems is you just plop a new piece into the ecosystem and you can look at how the system is going to react. You can understand how the system works, and then also you can see what's the potentials for adaptation in the future. So yeah, Phragmites is near and dear to my heart. I want to point out that invasive species are a definite product of the Anthropocene. The way that we define invasive species. So they're human caused, and then by virtue of their disruption, they're causing harm to things that we value. Impacts from invasive species are growing the more we account for them. And that's because there's more invasions due to human transport, and because systems are more invadable due to human disturbance. And then further, they interact with a whole host of other human-induced global changes like climate variability, nutrient enrichment, loss of biodiversity, and land use fragmentation. And these lead to reduced resilience of ecosystems and enhanced complexity and uncertainty in our ability to predict how ecosystems are going to adapt, what they're going to look like in 20, 50, 100 years. This paper that's being highlighted here, this Borgelt et al. From 2024, has kind of two main points. One is they quantify that invasive species are a bigger threat to biodiversity than climate change at a global scale. And they also highlight that introduction itself is the biggest risk. The math here is all done by tracking the routes of transported commodities worldwide and terrestrial alien species that are discovered in the imported country, but that were present in the exporting country in 1970 to 2019. And so you can see some of the ones here, particularly coming into our delta across the bay from Japan and from China. That's a fun paper to read. We're talking about invasive species because they affect so many things, and we talk about that in the future, but the truth is that golden mussel, for example, has threats, looming threats, but there's impacts already. There's impacts on recreation because the concerns over golden mussel introductions are causing reservoirs elsewhere in California to restrict the launching of boats that are coming from the Delta. So there's already things in play. We're already seeing impacts, and we're starting to just normalize it, but it's not, it's something that we have to address. And I think the next talk actually will discuss that sort of acceptance pathway. There's effects obviously on boating. I just mentioned fishing in terms of access and visibility, invasive plant effects on agriculture, duck clubs, other land uses are just, numerous. And, I just saw in the newspaper, we're looking at potentially at the federal side, shutting down Beltsville, the USDA efforts there's a big invasive species team in Maryland at the Beltsville USDA plant, and that's just another sort of hit that we're taking. Harmful algal blooms risk human health. They risk fish kills. The big one in 2022 with Heterosigma akishiwo, that was an invasive alga. Mute swans, finally we're starting to address them, but they are bullies. They are territorial. They take nesting sites. They outcompete native species, birds in particular, and then they also overgraze vegetation. And then lastly, Nutria, is everybody's favorite rodent to hate, but they have levee effects already. I mean, there's arguments that in 2004, the Jones Tract levee blue sky day failure, the levee failure was actually in response to burrows. I think the other point that I just want to highlight is that of all species that are problematic, invasive plants are just so ubiquitous right now, and they're so pervasive that we simply have to factor them into almost all of our water quality metrics. They have a whole host of water quality issues. You're just looking at some here in terms of that, slowing the water down, producing a lot of exudates and just making the water very poor in dissolved oxygen. That's slowing the water, then reduce sediment transport. And that can reduce turbidity, which a lot of native species like. Slowing water down also increases water temperatures, another problem. And then it creates habitat for non-native predators such as striped bass, the uber predator in our system now. And then, again, the cost of this is not, we're not really always factoring it in, but it reduces the effectiveness of all of these tidal wetland restorations that we're putting in that are required by mitigation for the state water project. For example, 8,400 tidal wetland acres are already, they're documenting the effects of invasive species on their goals. And again, with the cost of these impacts, if we don't recognize them directly, we're missing the boat because they're just growing. There's constant maintenance that costs money. There's risks for maritime shipping that's factored into decisions that are made by ships. There's risks for tourism, obviously, in terms of where you can launch your boat. Risks for property value, for a whole host of different impacts on your property. And there's very few success stories for eradication. I am still amazed that the Bay was able to hold back Spartina alterniflora invasion because there's such an empty niche there here. For, Spartina alterniflora being the cordgrass from the East Coast. That can't, once we have it out here, it's filling in those mudflats, but we know the mudflats are important for migratory waterfowl, and yet we're somehow have been able to hold that back, the invasion into that empty niche. Once plants arrive, really containment is our only option, and eradication is so very difficult, although I would love to hear all of the optimism that we're going to have this afternoon and potential some ways to get there. The vigilance it requires is too costly for any one agency. So this is an all of staff, all hands on deck approach that we need. And I also want to just really highlight citizen engagement to make that a reality. It's just eyes on the ground are so valuable. And so I wanted to say with all that threat, we do have some progress. Young and evolving estuaries like ours are imminently invadable. And we have a chance to keep some of the worst invaders from making themselves common. Risk reduction takes all manners of shapes, ballast water, treatment preventive chemicals, and barriers to settling, tracking using new tools like weekly repeat remote sensing or environmental DNA. And then perhaps our best tool, again, eyes on the ground and shared lessons learned through adaptive management through sessions like this. As well as, for example, the Adaptive Management Forum that happened earlier in October. I also want to highlight the Invasive Species Review by the Independent Science Board. It's from 2021, but it's still relevant today. Again, their recommendation is, quote, to encourage a more ecosystem-level, forward-looking, integrated approach to non-native species science in the Delta with specific consideration of climate change. And we are already at the Stewardship Council funding projects in that vein, working with landowners on Phragmites response, early detection, and rapid response recommendations with a directed action you'll hear about later today. And the last thing I want to highlight is that our Delta is evolving every day. Nothing is static. Non-stationarity is the norm. We have new invasives, new infrastructure, new restoration sites, new climate impacts, new regulations, mute swans, finally, part of the regulations. But tomorrow's Delta is always going to have invasives. And so the call to action is really to recognize that we have an advantage right now compared to 30 years ago, because so many have sounded the alarm. The Independent Science Board, the State of Bay Delta Science, the Synthesis efforts that have been written. And our challenge, our grand challenge, is to deal with prioritization. Which species to go after, which invasives, where, to what extent, and then this long-term problem we're going to have with perception of generational perception, shifting baselines. There's nobody in the Delta today that's never seen a Delta without invasives. And shifting baselines just means every generation there's a new normal that you accustom yourself to, and that you think that that's what it should look like. And so we'll always have this shifting baseline, and so addressing that is going to be another part of the response. And so again, looking forward to today, to hearing some science management interface ideas to strengthen our efforts with best available science. We're going to highlight research, tools, and management approaches, and hopefully we come through today with some good ideas for improving our response. Thank you.
Good morning. My name is Elizabeth Bersati. I'm a senior environmental scientist and adaptive management liaison with the Delta Stewardship Council, and I'm also one of the co-leaders of the DISC team. So we're going to start our first session today with management paradigms. And the goal of the management paradigm session is to show new ways we might approach and improve invasive species management in the future Delta and Sassoon Marsh. So the session will have 3 presentations, 2 in person and 1 recorded. Then we'll have a few minutes for questions after each presentation. And then at the end, we will have all the speakers up here for a panel discussion. And we'll also be joined by Rochelle Tanner, who's giving the recorded presentation. She'll be joining us live virtually. And so a few notes for the speakers. We have a timekeeper sitting up front. All of the slides have been loaded into one big presentation, so just keep advancing them. You don't need to open or close any files. We do have a clicker up here. Please make sure you speak into this microphone. That's so people online can hear. Let's see, and I think that's it. And so please welcome our first speaker, who is Abby Keller from the Department of Environmental Science, Policy and Management at UC Berkeley.
Hi, everyone. Like Elizabeth mentioned, I'm Abby Keller. I'm a PhD candidate from UC Berkeley, where I study decision analysis and quantitative ecology. And today I'm going to be sharing some of my work applying concepts from decision theory to help invasive species managers make better decisions when there's a lot of uncertainty. So this talk is in the context of the invasive European green crab, which is a marine species, so it doesn't actually live here in the Delta. But I think these results are kind of broadly applicable, and I'm looking forward to more of the talks today to get a sense of the commonalities. So just a kind of a quick background on the European green crab, it's commonly cited as one of the most impactful invasive species in the world. They're quite aggressive predators, so they consume shellfish in kind of large amounts that are often commercially and culturally valuable. Also, while they're foraging, they can uproot eelgrass beds, which is a really kind of valuable habitat for things like shorebirds and juvenile fish. So the green crab is here in California along the coast, but my research focuses mostly on the Northeast Pacific, so kind of Washington State and British Columbia, where for most of the last 20 years, the green crab has been present, but at quite low abundance. But in 2019, there was a bit of a regime shift where crab abundance really skyrocketed in multiple bays throughout the state of Washington. These coastal Washington bays produce 25% of the country's oysters. So this kind of rapid change was a huge threat to kind of the livelihood and culture of shellfish growers in the region. So the governor of Washington declared a state of emergency to respond to this kind of existential threat. But kind of the pace of change in these new circumstances were generating a lot of new uncertainties that were kind of straining the ability to make rational decisions. It turns out that managing the species is really difficult. So the region is fragmented by state, tribal, and federal jurisdictions. There's also a ton of private tide land. So we have all of these individual shellfish growers who are all making their own decisions about kind of if and how to control the green crab. But this kind of small spatial scale of decisions really doesn't align with the much larger spatial scale of the ecological process. So the green crab has this planktonic larval stage where the larvae move out into the open ocean for about 60 days. And during certain oceanographic conditions, they can travel hundreds of kilometers away from their source. So if you can imagine you're a manager of a bay, you have your local adult green crab population, you'll be removing the crabs, but they really easily recolonize from neighboring bays due to this long distance dispersal and influx of non-local propagules. So just to kind of build out this idea further, let's think about how a green crab population will respond to removal. So we can first consider a scenario where we have kind of an isolated population, mostly local reproduction. You can put in a high or low level of removal effort. The remaining adults will produce local propagules. Inevitably, there's going to be a few coming from somewhere else. And then kind of after density-dependent recruitment into the population, we see that the resulting density is closely coupled to the level of removal effort. So let's now consider a contrasting scenario. We have a much more connected population, many more non-local propagules. You can again put in a high or low level of removal effort. The remaining adults will produce local propagules. We have the same number of non-local propagules entering both populations. And then after kind of recruitment into the population, we see that the resulting density is now decoupled from the level of removal effort. So this project was part of a larger structured decision-making effort where I interviewed a bunch of decision makers throughout the region to get a sense of the types of decisions they were making and the challenges they were facing. And for many of these decision makers, controlling the green crab feels like this Sisyphusian task. They're wondering if their removal efforts are futile and if they're just going to be sinking money into this problem forever. So that these questions and this way of thinking are increasingly common in 21st century ecosystem management, where managers are often navigating these big ecosystem transformations that can benefit from considering broader objectives, so kind of beyond the traditional focus of just resisting change. So this way of thinking is really embodied by this decision framework called resist, accept, and direct. And we found that this framework was really applicable in this context. So here we're framing the problem of green crab management as this choice between resisting change by kind of continuing to remove the green crab with the goal of conserving historical or current conditions, or alternatively kind of accepting that this is kind of an invasion trajectory we can't control. And we should instead invest resources in adapting to a new normal. But for green crab, it's really hard to know whether you should be resisting or not. There's a lot of uncertainty around things like rates of population growth, colonization, and even the effectiveness of removal is not well characterized. So here comes in decision theory, which is a really useful way of thinking when trying to make rational decisions when there's a lot of uncertainty. And I see kind of decision theory as being two main things. The first is trying to help us understand how to kind of break down and describe a decision problem. And the second is kind of a set of methods and formulas that we can use to optimize decisions under uncertainty. And one of these methods uses a Markov decision process model that's useful for sequential decisions. So by sequential, I mean kind of an annual decision about how much effort to put into removing green crabs. You're making this decision every single year. So this type of model takes an action. So here again, this is the annual removal effort. It also takes a state. So in this context, this would be the density of kind of the local green crab population that a manager is managing. So let's say today we're at time t. The system is in state A. You could take an action. That action causes the state to transition to a new set of states with different probabilities associated with these transitions. And again, this process continues where you again take an action, which causes the state to transition again and kind of so on forward in time. You can imagine doing this for 100 years. So what we're looking for is the action to take today, considering all of these kind of trickling and cascading ways that today's decision could influence the future state. So we first built a stylized model of green crab population growth, colonization, and removal. And here I say stylized because I mean that this model is relatively simple, where we're just focusing on the main processes and using parameter values from the literature to inform these processes. And we're accounting for stochasticity and randomness in these rates. So for example, the colonization rate can vary dramatically from year to year due to things like changes in ocean temperature or kind of big oceanographic events like El Nino. So we can use this model to quantify how a population responds to removal and relate the local equilibrium population size, which is here on the x-axis, to the green crab removal effort or action, which are I'm going to be the colors in the next few slides. And since we're kind of accounting for uncertainty in various parts of this model, there's going to be some probability associated with the equilibrium population size. So we can again first think about a regime where the colonization rate is low. So this is an isolated population, very few non-local propagules. If you did nothing, or if you applied a removal effort of 0, the population would reach carrying capacity. So that's kind of K on the x-axis. And as you increase the level of removal effort, we see that the population is responding to removal. So, a higher removal effort equals lower population size. So again, this kind of different regime where the colonization rate is much higher, you have a much more connected population, many non-local propagules. We see that as we increase the level of removal effort, we really can't knock back the population. So this is a scenario where kind of the population size is decoupled from the level of removal effort. So the question then becomes, when do we resist and when do we accept? So we're thinking about this as a multi-objective optimization problem, where we're looking for the action or the optimal removal effort that minimizes both ecological change and the cost of removal. So here the cost of removal is a function of the removal effort, where kind of lower removal efforts have a higher value. So essentially, it's more costly to have a higher removal effort. And whether this relationship is linear or nonlinear will depend upon the constraints of a decision maker. And the amount of ecological change is a function of the green crab density. Lower green crab densities have higher value. And kind of the shape of this relationship likely depends upon the impacted species. So whether it's kind of the rate of predation on shellfish or kind of the rate of degradation of eelgrass beds. So since this is a multi-objective problem, we have to be weighting these two objectives. So first I'll share results for a scenario where a decision maker cares much, much more about minimizing ecological change than minimizing the cost of removal. And before I get to the results, I just want to clarify a symbol that you'll see. So this kind of decision boundary between resistance and acceptance is really sensitive to kind of the colonization rate or the kind of size and level of connectivity between a source population and a local population. So if alpha is 0.5 K, this means that the source population is on average half the size of the carrying capacity of the local population. And if alpha equals K, it means that the source populations are equal in size to the carrying capacity of the local population. And because we're kind of in this model, we're accounting for kind of randomness and stochasticity and the size of the source population and the level of connectivity, this alpha is kind of a summary statistic of a probability distribution. So here, kind of a preface to the results. On the y-axis, we have the optimal removal effort. On the x-axis, we have the local green crab density. These three panels moving from left to right correspond to increasing levels of connectivity between the local population and source population. If alpha is really low, it's kind of this isolated population scenario, it's not going to surprise you that it's optimal to resist. So here, kind of, it's best to just continue throwing all of your resources at the problem. But as alpha increases, the local population becomes more connected to source populations. We see this switch between resistance and acceptance, where at high local green crab densities in this kind of region in the dark gray becomes optimal to accept. So this is a removal effort of 0. And as alpha continues to increase, the removal effort is 0 for all local green crab densities. This means, the best strategy is to kind of accept that this is an ecological trajectory you can't control, and instead invest resources in adaptation. But really, this question of whether to resist or accept is a reflection of the values of a decision maker. And the way that the decision maker expresses these values is how they weight these two optimization objectives. So previously, we considered weights where the decision maker cares much, much more about ecological integrity than minimizing the cost of removal. But we can kind of do a similar exercise and see how these optimization results change if these two objectives had equal weights. So just for comparison, these results on the top were kind of the previous ones with the high objective weight for minimizing ecological change. So we can see these new results for these new objective weights where these two objectives are weighted equally. And when alpha is low, so this is again a loosely connected population, it's still optimal to resist. So a kind of a removal effort greater than 0. But now as alpha increases, so here this is saying kind of the source population is on average half the size of the carrying capacity of the local population, becomes optimal to accept for all local green crab densities. So kind of a removal effort of 0. And as you can imagine, as alpha continues to increase, it kind of is similarly optimal to accept for kind of more connected populations. So you've seen that kind of the way that you formulate the objective function and the way that like the decision makers' values are expressed in the objective function really changes the results. So we built this our Shining app, this interactive web tool where decision makers can specify their own objective weights, build their own objective functions, and kind of run these same optimizations for themselves to see how the results change when they are able to express their own values. So I just kind of want to conclude with kind of some brief thoughts about how I see a model like this informing actual decision-making on the ground. So, you know, like all models, this is a simplified version of reality. So yeah, I describe this as a stylized model. So we don't necessarily see these results informing the very kind of like detailed, granular day-to-day or annual decisions that decision makers are making. But we really see this kind of as a tool to build intuition about the problems, kind of to understand whether a population is kind of squarely in the resist category or in the accept category. And something that's been really interesting to see is as we've shared these results with decision makers, we've seen that it's kind of really been used to kind of build conversations around how the problem of green crab management should be framed. So we see this kind of as an object to create consensus that kind of the problem of green crab management is really this choice between resisting and accepting. So that's been kind of a useful outcome of these results. Yeah, that's it. Happy to take any questions. So the question was on the first slide or one of the earlier slides, there was a big boom in 2019. I guess a couple things. I think the kind of level of connectivity is really sensitive to things like El Nino. So I think that was an El Nino year where we had kind of there's tons of green crab in California and Oregon. So they transported up probably at that point in time. But also those bays, there's a lot of, they're really kind of insular. So probably it's kind of just a lot of things happening all at once. So just kind of creating the right, like a window of opportunity, creating the right conditions where, you know, a big larval plume just settled. And yeah, so I think that probably El Nino. Yeah, the question was, how did the crabs get to the West Coast in the first place? So the first point of detection was in the San Francisco Bay. I think it was just trade shipping.
Regarding the time window before, well, the determination of when you go from resist to accept, is that usually within a 5-year window, 10-year, 20-year? Because it seems along with all invasive species that if you go through the cycles of them not being able to either for plants like reseed an area over and over again, and eventually all the seed bank ends up kind of germinating and not repopulating and replenishing itself. I would think that would go along with the crabs. So, you know, the eggs that are out there kind of come back in, but those smaller crabs that are babies aren't able to reproduce, and then you remove those, and eventually you can kind of whittle them down. So is it usually like a time frame that you determine when it's just the accept period?
Yeah, that's a good question. The question was, is there a time frame for when you should be considering accept versus resist? I feel like with green crab, it's more of a question of space rather than time because the dispersal is so long, I think, and I imagine relative to plants. I think the thing that's challenging with green crab is you have a huge El Nino event that just dumps a bunch of larvae in your area, and then you kind of remove them all, but then you have kind of like a few years where things are a little bit better. But then like 3 years later, you get dumped again. So I think the time frame kind of probably corresponds to the patterns of El Nino, which are increasing in frequency and magnitude, of course. But I think it's really a question of space. So rather like how isolated it is versus how, yeah, like a question of space than time.
Currently, there are 2 objectives to weight. How would this framework be used under more than 2 objectives regarding weighing these objectives?
Yeah, so I mean, the way that the different objectives.
Y'all repeat the question.
Okay, repeat the question. So the question is, if there's more than 2 objectives, how do you incorporate that into the decision problem? So the way that the 2 objectives are kind of incorporated is just like a linear sum of these 2 objectives. So really, you can very easily add an objective. The question is how you develop the weights. So they sum to 1 and they're kind of arbitrary and a little bit abstract. So there's kind of some useful tools kind of from decision science that you can use to kind of probe decision makers to help them develop these quantitative weights for kind of their more like abstract values. But yeah, I think it's easy to kind of add more linear weights.
I was wondering what kind of information in terms of, understanding of carrying capacity. What should land managers and people on the ground, scientists that are studying these, what sort of metrics should they be measuring in order to inform models that they are going to show?
Yeah, so the question was, what information does a manager on the ground need? So things like carrying capacity when they're trying to apply these methods. So definitely there's a lot of like assumptions about, this is decision making under uncertainty, but also still assumes that you like know some stuff. So I think some of that type of intuition around, so something like carrying capacity, understanding is the population increasing or decreasing over time. But something that's really important in this space, there's a lot of larval tracking, larval modeling efforts. So that's really useful for understanding kind of the level of connectivity, which is kind of really the most important factor in this problem. So yeah, basically the people use these big regional oceanographic models where they'll drop a particle somewhere and see where it ends up. So using the results of those models is going to be really critical for people on the ground, knowing how connected their population is to others and whether they should be accepting.
Really interesting topic before. I'm just curious about the way that resistance and acceptance is framed. It's almost like a zero-sum binary, where it is either acceptance or it is resistance. And you nodded to this in kind of the real world elements of like the qualitative elements, whether the real world just entertains a little bit more than that. So how does your research entertain the kinds of that line between acceptance and resistance in a way that kind of decouples it from like the zero-sum reduction?
Yeah, that's a good question. So it was the questions about how this kind of binary choice between resist and accept, and how do you incorporate the more of the nuance associated with decisions on the ground. I think something that we see in these results, so just the outcomes of the optimization tend to be like do everything or do nothing. That's just kind of like we saw that very clearly. And I think in general, we see that people are just going to get as much money as they can for controlling the green crab. And then it's like, should you spend it all or not? Like, I think that it really kind of in practice tends to be this binary question. But part of the resist accept framework, there's also this other option of direct. So that is kind of like, you're going to direct the ecosystem to a new state. So something, we didn't really consider that pathway in this analysis, but something that like that could be, we're just going to be removing green crab near resources that are very valuable, and kind of in other areas we don't care as much. So I think, future work could be considering that aspect of space and thinking about where we have valuable resources to kind of take account for the nuance on the ground.
Thank you, Abigail.
And there may be a chance for more questions when we start the panel after the next couple of talks. So our next presentation is by Brett Milligan and Elsie Platzer from UC Davis.
Good morning, everyone. I'm Brett. This is Elsie, and excuse me. We are going to take you through a very quick exploration into our project, Just Transitions in the Delta, which is looking at adaptation to increasing salinity in the Delta and climate adaptation generally. So I'm going to provide an overview of the project quickly, and then we're going to dive into some conceptual models of what we think might happen ecologically under some of these features. So real quick, this is our team. Elsie and I are one of some 25 people working on this 4-year research grant. It's a multi-campus UC research grant. And as many of you probably know, salinity is a very complex phenomenon in the Delta. It's dependent on many different factors, whether that's policy, environmental change, climate, technology, what infrastructure is put in place, land use, water. So that's some of the things that are underneath that in terms of speaking about decision making under uncertainty. So we're not going to go through all these all right now. This is just this is basically a participatory scenario planning process, and we are trying to take all of these factors in to thinking about some potential adaptations to this problem. Title says just transitions. What is just within this context? Generally, just transitions refers to trying to make a just transition from where we currently are in ways that are perhaps more equitable. So for our project specifically, we're looking at future salinity management and climate adaptation in the Delta, and how might we make that more equitable. And this is our overall research process. Something's happening weird with that slide, but so overall, we're doing a participatory scenario planning process, which scenario planning generally means you run multiple scenarios in parallel and compare and assess what you get from those. We're very heavily involved with public engagement and trying to include those who might not normally be in on these conversations, tribes, environmental justice communities, and others. And the goal here is that we can build shared learning and visioning through having that community help us build these scenarios, then modeling and run those scenarios to see, what benefits and impacts happen across them. And so our hypothesis in this project is, can we build trust and capacity through this type of scenario planning process? This is a figure you'll see in scenario planning. We're trying to look at a range of possible futures, not the most plausible or possible, but what's also possible. So kind of that larger range rather than just status quo or the limited range of what we normally might do. And so with that, I'll jump into our 6 scenarios that we have that we're modeling in this project. And so the first is business as usual. This is exactly what it says, business as usual. We keep trying to maintain the X2 standard into the future, which I didn't mention this, it's perceived it's going to be much harder to keep the Delta fresh in the future with climate change. You're going to have precipitation patterns changing, higher sea level rise, which means more higher tides coming into the Delta. It's going to be increasingly hard to maintain the Delta the way we do. But we look at this into the future. Can we keep doing that? To what degree? For how long? A tunnel is the Delta Conveyance Project, which is basically the Bethany Reservoir alignment. We didn't plan on modeling this. A lot of people wanted this modeled in comparison to the other scenarios. Eco Machine is trying to strategically use restoration to literally try to move where salinity goes. So this is a couple spots we're looking at here. This has been shown in modeling to work in Suisun Marsh, where if you open that up to tidal action, it actually pushes some of that salinity away from the Delta. The other one is Frank's Tract. Some of you may be familiar with Frank's Tract Futures, which was a multi-benefit project of building up land masses there to serve as habitat while also literally being a barrier for salinity going in there to then where it takes it down to the pumps. So to what degree can restoration actually help with salinity? Our New Green Watershed, this is basically looking at restoration much more systemically and broadly. And this was from a lot of tribal input and others and also people who said, look beyond the Delta and upstream. But this is looking at a holistic strategy that recognizes that subsidence is an issue in the Delta as part of salinity, because if the levees break, you get salinity coming in. So this is a strategy to basically keep all the soils wet in the Delta, restore wetlands where you can, and on the edge. So basically, I won't go through this whole plan, but holistic restoration. And this is not just in the Delta, but also across the entire watershed. Where can you best manage uplands, everything upstream of the Delta, which we technically think of these as water infrastructure now, but where and how would that be best, such as restoring mountain meadows, land fire stewardship, that you're changing, how much tree cover you have and how all that might affect where and how water moves down into the Delta, as well as flood protection. Bolster and fortify, this scenario was really looking at what kind of gray infrastructure can you put into the delta, gates, barriers, operable gates, things like that to try to attenuate salinity. And we looked at all kinds of options for this. What we've come down to is a through delta corridor. And if you fortify that corridor through the delta, what do you get? What are the trade-offs? A lot of people are really interested in this versus the tunnel. So this is another one we're looking at. And then lastly, calling on reserves. This one, the adaptation isn't in the delta, it's rather upstream of the delta and looking at how you operate reservoirs. Water releases have a huge impact on how much flow is coming through the delta, which in turn impacts how much salinity, where salinity is based on how much outflow you have. So looking at smarter, better ways of managing reservoirs. So that was a whirlwind tour of our scenarios. For all these, we're doing common stress and sensitivity testing. One of those will be tiered inflows, outflows, like how much flow is coming through the delta and how that varies across the scenarios in terms of how well they attenuate salinity. We'll be testing them against a long-term drought. A lot of people asked us to break the delta, around 3 years of drought, something like that, where you run out of supply to actually attenuate salinity. And with that application of climate projections, sea level rise, warming air, water temperatures, and if we can, modeling some potential levee inundation events. So all the scenarios will be tested against that. And so we have like 6 different criteria that are, again, publicly sourced of ways of evaluating these, from recreation to economy. But one of those is ecological modeling. And that is what Elsie is going to talk about.
Thanks, Brett. So first, I'm going to take you all through kind of the near-term future effects of different scenarios and how that would affect the ecology of the delta. So something that all of our scenarios have in common in the near term is that there will be continued management of the delta as freshwater to ensure that exports can still happen. But we're interested in looking at how, even under this common condition, the scenarios will vary. So looking at just 3 of our 6 scenarios that are, I think, pretty distinct. Brett did a good job of foregrounding those. We have business as usual, which is kind of following current trends as far as we can. New green watershed, holistic restoration for salinity attenuation, for food web benefits, for subsidence reversal, green economy, indigenous sovereignty, all kinds of great stuff. And then also bolster and fortify with that focus on gray infrastructure and kind of hardening the landscape. So let's look at the effects of business as usual. Looking at how current trends are playing out in the delta, we would only expect those trends to continue and accelerate. So we would see a decrease in turbidity, because that's already been happening with SAB and with fewer flows, as well as an increase in temperature, especially water temperatures due to climate change. And both of those things are beneficial for freshwater invasives, whether that's SAB or the fish that love to live in SAB, including bass species and sunfish. Now, thinking about new green watershed instead, all of the restoration that's taking place would, of course, increase habitat availability for native species, and also hopefully plankton production with things like restored floodplains, operation of managed wetlands, floating wetlands, wet soil agriculture, etc. But at the same time, tidal restoration in Sassoon Marsh to attenuate salinity would have the effect of decreasing tidal extent coming into the delta. So the positives of this scenario would be that increased plankton, increasing the food web, and increasing habitat would benefit anadromous fishes that we love, like salmon, whereas decreasing the tidal extent would increase the risk of upstream HABs, as the upstream reaches are going to be more stagnant over time. Now, taking a look at bolster and fortify, obviously, when you harden the landscape, there's going to be a channelization of waterways. Living levees kind of turning into hardened levees will have that effect. And that will increase water velocity as water is pushed through quickly. That will also decrease habitat availability and accessibility for species. And of course, as water velocity increases, water residence time decreases, leading to a reduction in plankton production. And for forgot about the fishes. For the decrease in habitat availability and accessibility, obviously that's going to have a negative effect on the salmon anadromous fishes that rely on those habitats. So now I'm going to toss it back to Brett to talk a little bit about the long term and what these scenarios would look like played out far into the future.
That was fast. You sure you covered everything you wanted to?
Yeah.
Okay, so again, I want to highlight this point of the further we go into the future, the harder it's likely going to be to maintain the delta fresh in the way we have for the last, 80, 100 years. These are a couple modeling results. On the left, you see a schism model of what happens with 1.2 meters of sea level rise. We can debate when we might hit that level. But it shows, you can see where it goes towards the green and the yellow, that's showing increased salinity as it comes into the delta. So again, you're going to get these higher tides and salinity penetrating into the delta unless you have more water to throw at it upstream to bring it down. And then on the right there, we're just seeing a model exploration of what if you reduce exports. I think this was like a 20% reduction. I can't remember offhand. But these are 2 points, Emerton and the Banks pumping plant. And you can see that if you reduce exports, you get greater outflows, which is what you need. Bottom line, this is from the Delta Conveyance EIR, and it just real quickly, it's showing you just how much exponentially more water is required to keep the delta fresh as sea level rises. Those are in million acre feet along the bottom. So with that context, over to you.
So let's imagine this long term as a pseudo seventh scenario that I've called the salty or saltier delta. And under this scenario, salinity management in the delta becomes untenable long term, essentially. You'll see increasing cycles of drought and flood, extremes becoming more common, lots of uncertainty, and adaptability is the name of the game in order to survive. So when we take this new boundary condition to be true, freshwater cannot be consistently maintained in the delta. The big result that you see is an increase in variability over space and time. So fresh, salt, brackish, these conditions are going to fluctuate a lot. What that means is that water exports may periodically be interrupted or stop altogether. If the delta is salty, water can't be sent. And what effect that would have on flows is really a question mark because, we don't know how extensive those periods of stoppage would be. We don't know when, and we don't know how much upstream flow would kind of be allotted to offset that. So that's a really big uncertainty. But then thinking about the effects of variability on the species composition of the delta, what we think would happen is that freshwater invasives would suffer under these conditions, whereas estuarine adapted species that tend to be our native species would actually benefit. So if we're thinking about, in particular, our native fishes, these are 3 native species that we have, tule perch, split tail, Sacramento perch. They all have pretty extensive salinity tolerances, up to 19 ppt. Whereas our non-native freshwater invasives, like sunfish, bass, catfish, they have very much smaller ranges of salinity tolerance. So under a saltier delta scenario, we would actually probably see a big reduction in freshwater invasive species. And how do the long-term outcomes vary between scenarios? Just really quickly, even if we can't maintain freshwater in the delta long term, we do think that a scenario like New Green Watershed would be less vulnerable to catastrophic shifts because there's been some resilience planning put in place with restored habitat, doing some subsidence reversal so that the risk of extreme flood events or levee failure is a little bit less drastic. Whereas something like business as usual, there hasn't been as much mitigation in place. So one catastrophic event could lead to a huge change on the landscape. And we still have a lot of questions going forward. Obviously, we're thinking about things like, is a saltier delta a more resilient delta? Which adaptive scenarios serve us best in the near term versus the long term? And who are the winners and losers in each scenario? And that's just a little taste of our project. And now we're out of time. So if you have questions, please ask. I do have time for some.
We have time for questions. Just remember to repeat the question into the microphone so people online. Yes.
Regarding the salinity, I'm curious on if there is a historic pattern of the salinity of the delta increasing and decreasing over millennia. And if so, is it thought that it might help with the possibility of the invasives that have tended to overtake not just the living animals that are there, but also the plants, because we understand that a lot of invasive plants are averse to salinity. Since it's such a big catastrophic effort to eradicate them manually, whether it's with spray or whatever, that maybe it is part of the natural ebb and flow of having salinity kind of increase upstream, kind of wipe out all of the non-natives, and then it reduces over again, maybe a decade or decades of that as a natural process.
To repeat the question, it was a long question. Basically, I think you're asking about the historical salinity regime in the Delta. Yes, okay, good. Historically, in pre-colonial times, the Delta was variable based on water year, based on flows. So it wasn't kind of a long-term cycle as you were maybe suggesting, but more like year to year, it might be brackish or it might be fresher, just depending on the climate conditions. The maintaining of a strictly freshwater delta is a new phenomenon, and that's for the benefit of water conveyance. So the species that are adapted to an estuarine system that is variable in salinity would be able to weather these changes, whereas freshwater invasive species, including SAV, would not be able to weather those changes. So yes, I think part of your question was also asking, would freshwater plants like SAV or water hyacinth suffer under that salinity regime? I think they would, but it would have negative consequences on the human aspects because so many people rely on water exports. Yes. Tag him.
So I think the key thing there is like when and how you might let it vary that you get the benefit. And I think also the baseline is really tricky. I mean, there's a lot of debate of what, like, was the delta always fresh? You'll see some of these old diagrams from like going back to the 19-teens that show, oh, look how much salinity came in, but that's not taking into account how much was already being diverted. So it was already a shifted baseline that was being looked at for that. But then there's a lot of stuff that shows that it really was primarily stayed fresh. But that was also because structurally it was entirely different. It was spongy connection between riparian habitat, floodplains, and all that. So it's just so radically altered. So I do think the baseline thing is a little bit of a tricky one.
There are 10 invasive species in families of CFA. So if you become saltier, I don't know that you're going to replace species that are invasive. You might have different species for that. I mean, maybe your plants and the fish that you're used to and the delta that cause problems, but I don't think it's going to actually decrease the number of invasive species in the delta. It will just change which ones we have, unfortunately.
You could repeat that.
So the question that was more of a comment was that there are also invasive species in the bay, which is salty. So just a change in the salinity regime wouldn't eradicate invasive species. And that's definitely true. I think it would threaten the freshwater invasives that we have and we struggle with currently, like SAV and sunfish and all of those types of familiar characters. But it's true that a new suite of invasive species could come along and establish, and we just don't know.
Great thought. With what you guys were showing there on the new green watershed modeling there, talking about restored habitat, what habitats were we talking about for restoring riparian, saltwater marsh, freshwater marsh? But I feel some of those obviously would help a lot with the situation more than others in terms of the impacts it can provide for median. So is it just really all that habitat or more specific habitat types for that funding?
So the question was about our design for new green watershed, the scenario and kind of which habitat types are included in restoration. And we've actually been thinking an extremely large amount about this, and there have been many, many iterations of different maps and different designs, reviews by expert panels, by tribal panels, and all of those kinds of things. And so we do have kind of a general mosaic of different habitats that are assigned to tracts, and we have thought about what the interactions between different habitat types would look like. We have a mixture of tidal wetlands, levee setbacks, riparian enhancements, floodplain restoration. We have thought about floating marshes, carbon sequestration, rice fields, operation of managed wetlands, all kinds of things are incorporated within that scenario. And if you are interested, you can see kind of a breakdown of this, I think, updated on the project website. Maybe it's on the project website.
I need to update it again.
Soon it will be updated on the project website. Yes.
Thank you. Thank you. And our recorded presentation is going to be by Rochelle Tanner from Chapman University.
Hi, everyone. Thanks for having me today. My name's Rochelle. Sorry I can't be there with you. Today's the last day of class for us, but I'm excited to share some of the results from this 5-year study that we've been doing in Sassoon Marsh. I'm sure I don't need to remind you all, but Sassoon Marsh is a wetland on the edge of the California Delta next to the San Francisco Bay. It has a big hand in protecting the Delta from saltwater intrusion, but it's managed by a group of about 120 landowners that are mostly private duck clubs. These red areas are land owned by DWR and Cal Fish and Wildlife, but all these tiny little parcels are duck clubs. And increasingly, this area is overrun with Phragmites. This figure is from a 2023 paper by my colleagues at the Sassoon Resource Conservation District, and it showed that Phragmites has increased by 234% in the last decade and is only continuing to increase. And so this is a really big management problem for the SRCD and the landowners in this area because they don't receive federal funding to manage their land or the levees that protect our water supply. This is Phragmites. If you have not had the pleasure of being stuck in it, Phragmites is something that can't be contained to a particular land parcel. And so when we think about invasive species management here, we're thinking about how to engage multiple landowners in collective action towards the management of the species. This isn't something that the SRCD or anyone in this area had thought about from the angle of how do we use social science to address this problem. This had been primarily kind of a biology or a physical issue up until this. And so in this project, we not only looked at the biophysical aspects of what are the best practices in managing Phragmites, we also looked at the social and psychological dimensions of how do you get people to work together who don't really have that baked into their management plan. So I'm only going to talk about the social science. These are some of the data sets that informed the social science. So in our expert elicitation, experts emphasize the importance of continued communication and early detection, or EDRR, which you guys are probably familiar with. When we evaluated who trusted messengers would be, we found that they needed to share cultural values with their population, whether this was their livelihood, their background, hobbies, political views, age. I'll show a slide coming up, more detail on that. We also found that they should have moderate management power, so they should be able to distribute resources, but not be the source of those resources, because our landowners very much preferred low regulatory power. And this was confirmed by the expert elicitation. This is the best way to have someone be the figurehead of a collective management scheme. The experts also said it was really important to show policymakers and managers firsthand damage in the field through walking the levees, finding long-term funding options for engaging landowners beyond initiatives, right? So like this project was only 5 years and we didn't even have funding in the project to do the invasive species management. And so, applying these findings is really important and it requires a lot of funding. The experts also critically underscored the importance of public outreach and garnering support and literacy for invasive species control. I'm going to come back to this after I go through the results from the main study, because this became a really big focus of the work that is coming out of this and what we're continuing to do. So it may come as no surprise that a bunch of duck clubs are really invested in protecting waterfowl habitat and waterfowl hunting as their primary land management goals. This is part of the survey that we distributed about values about invasive species management. So cultural values that they felt were most important for their connection to their land. And what we found was things like managing for unique habitats, being a responsible good steward of the land, protecting through responsible management, this acknowledgement of interconnectedness between both property owners and the environment. And this last one, which was no surprise, protect ducks. Of course, they wanted to protect their ducks. We probed a little further into how they were applying these cultural values to the way that they manage their land, and we came up with some interesting but maybe a little bit concerning trends. We found that managers identify invasive species by their impact on waterfowl habitat, which is not necessarily what makes an invasive species, but more concerningly, they said they felt familiar with invasive species, but they mostly relied on the Sassoon Resource Conservation District to tell them that something was invasive. So this is funneling information through one actor. They're not making decisions on their own accord or based on their own literacy of the issue. They're really relying on one entity to tell them what to do, which could be good for collective action, but it is not so great when you think about all of these actors acting independently before you have a collective management scheme. We did a lot of work on looking at what kinds of language would persuade a landowner to do something about Phragmites on their land. There's a really large body of literature that shows that positive emotions and action-oriented language that avoid sociopolitical conflict are the most useful in persuading members of the public or non-scientists to do something about an environmental issue. And so we followed up on that body of literature in this specific case study and found exactly that. These are things like habitat recovery, collaborative approaches, protection of unique habitat, working together, stewarding the land, resource managers. Those are the things that tested the best for getting a landowner to adopt a collective action plan. Now in this slide, I'm showing that we looked at whether Delta researchers and managers outside of Sassoon Marsh would be able to identify this type of language. And they were less than 50% accurate in identifying what would motivate a landowner to do something about an invasive species on their land. And so this is an area where we need a lot of education, both within our ranks, but also with landowners, to kind of have a common language that we communicate about invasive species in. We further probed into how landowners got their information because we were concerned that they were just relying on the SRCD for everything. And so we used a framework of these knowledge frameworks to kind of look at where they were most reliant on outside sources of information. Some of these are probably quite familiar to you. We looked at a lot more than this, but these are the three that I want to highlight today. Primary knowledge acquisition is very low uptake in this population, which is not surprising. This is, reading peer-reviewed literature and reports. The SRCD has water managers, and these water managers are the ones that have the direct contact with the landowners pretty frequently, at least once a week. And most indicated that both bidirectional and unidirectional means are common, meaning they both communicate back and forth, but also receive information from these water managers. Many landowners reported too much information this way. They couldn't handle the flood of information that they were receiving. And they also preferred in-person communication. They did not like online forums. They found them inaccessible. Even things like emails. When we distributed these surveys, we actually did most of it by paper mail. And part of that is because the median age of this population is 67. But that's something to keep in mind when we're talking about collective action in this area. Knowledge application was the highest use framework because it meant that they were taking something that they learned from the SRCD and using it in the management of their land. Other types of knowledge application like advocacy and public outreach scored very low, but again, even this population underscored the importance of public outreach in garnering support for these kinds of management activities. Now, this is the only bit of results you'll see from the whole study that we did. We did a spatial prioritization model where we found that when you only weight the biophysical dynamics of protecting managed wetlands from Phragmites, you get this kind of distribution pattern where you have high concentration of resources in public lands. But when you reweight that to look at the social dynamics of things like neighborly behavior and parcel size and whether landowners are willing to share resources across these boundaries, you see a complete shift towards the western side of Sassoon Marsh in where you should spend your resources and where Phragmites control will be most effective. And so I think this extreme comparison really shows that when we take into account the social dynamics of resource management, we have a very different way that we should distribute those resources. And that's something to keep in mind when we're doing future plans for managing Phragmites in Sassoon Marsh. Okay, in a nutshell, what we found was a local non-regulatory agency with moderate power to distribute resources like the SRCD should communicate to landowners about collective action measures. SRCD is the only entity in this area that fits that description. So that is a concern, a vulnerability in this communication framework. They should communicate about removing invasive species in the context of ducks and waterfowl habitat. I think that was not very surprising, but good to document. The wording is really important for communicating about this. So things like coordinated plans instead of laws and regulations, stewards of this land instead of habitat maintenance. There's a large list of that. And what this means for broader invasive species management is that when we only draw upon biophysical information to prioritize invasive species control and the distribution of resources, it's completely opposite, at least in this case study, to how the social dynamics play out in terms of who is willing to work together to manage the species across parcel boundaries. And so that means a lot for the likely success of the distribution of those resources, and that's something that we really need to keep in mind. Okay, so I said I keep talking about public outreach, and I wanted to talk a little bit about that because we did actually do some public work because this was such a big theme that came out through all of our social science studies. So this is the project team that worked on the social science for this, this project. Clearly, the landowners trusted us, but how do we know that the public will also trust this group, or who does the public trust at all? Now, I know, there's some question of whether the public should be involved in invasive species management, and I want to try and convince you of this here. First of all, the expert elicitation we did with experts across North America said that it was necessary. So all the experts say that we should do this. When you do early detection, rapid response, it's nearly impossible to do it without a quick turnaround and eyes on the problem at all times. Who is out there all the time? The birders, the people who are boating, the people who are fishing. They're out there much more than we can be. We think the public wants to be involved, and this survey confirmed that the public does want to be involved in this process. And there's two good things about this. One is that responsible management involves all affected parties, especially on public lands. And two, participation in natural resource governance will improve public science literacy and support for other initiatives that we want support for. And I just love this flyer from Calfish and Wildlife. Their wanted posters for some of their invasive species, very cute. I think we need to be looking into the efficacy of how we engage the public. And so we put out a limited survey in California and Florida to target the Delta and the wetland and the Everglades in Florida with 800 people. And so I'm going to show you some of the results from that. We found a mismatch between the perceived responsibility and reporting and management and the trust of these frameworks. This is unsurprising to those of us working in this area. We know that the public doesn't trust the government to do something with the information that they are given in a timely manner. What those are showing is this is who the public thinks should be controlling invasive species, who they want to report it to, and then who they trust to actually do something with that information. In a perfect scenario, this would be a flat line for any of these, and preferably the top one, right, which is in this case, the state government. But what we're seeing is that even though the public thinks that, the state and the federal government should be responsible for doing something about this, they don't trust them to do anything with the information, even if their public were to report it. These drop-offs here, right? The federal government one, which is this, is particularly bad, this yellow one. So there's this mismatch, and that's something that we need to address and repair in the way that we pursue future public engagement. One way to repair this relationship is by using values-based communication, which is clearly a through line of a lot of the research that I do. This is a replication of the Duck Club responses, and then this is the public survey. The only reason there are more in the Duck Club responses is because we asked specific ones about property owners and ducks, and clearly the public doesn't know anything about that. But otherwise, this is the same survey question that we asked both populations. They are largely in agreement with our Duck Club owners that the top 3 values for managing invasive species are: it's important to manage unique habitats, it's important to be responsible stewards, and it's important to protect our lands through responsible management. And so using this kind of language can kind of open the door to a trusting relationship if the appropriate actions are followed through this language. So in practice, what does this look like? We asked the public, how would you report an invasive species, just as like a first pass at this? And they said that they would prefer to report it through a social media platform, which is really interesting. There wasn't a huge divergence though in the options that we gave them. We talked about paper forms, we talked about email addresses, all of that, but what came out on top was like a social media platform. And even more interestingly, they said the state government should be the one to receive this information and manage the invasive species along with the federal government. This reflects another misalignment in how people think invasive species are managed. We know that it's done on the local level, but they seem to not realize, all of the actors at play in the management of species that are in their public recreation areas. I found this really encouraging. Only 7% of respondents, so 7% of 800 people, deemed no government entity trustworthy for reporting. So this means that there is a willingness to trust the government to do something about it, but that trust is being broken right now. And only 2.9% of respondents said that they don't need to do anything if they see an invasive species. So they want to be involved, but they just don't trust the current pathways of communication that we have right now. So our next challenge in EDRR engagement is figuring out how to get the public on board with this. And very paramount to that is, how do you engage a population that has no connection to the issue currently? When we apply for grants for this, the question we get a lot is, have you talked to the community groups about this? Which is something we're really sensitive to. That's something that we do a lot of in our other work. The reality is there are no community groups that have this as one of their main charges or part of their mission statement, because they don't see this as a public issue, and we need to change that. Christine and I, my colleague who we're working on this right now, we're looking to do some experimental manipulations in recreation areas that kind of look at how the public responds to invasive species, whether those are real species or things that we put out there that we want them to see. And we think, after doing this sort of experimental manipulation and monitoring, we need to host community literacy workshops to get people engaged in this problem, and technical info sessions for managers who will handle reports to kind of let them know what the public is looking for from them. So if you're interested in this sort of thing, we haven't started yet. So if you want this on your parcel of land, or you know where we could get funding, send me or Christine an email right there. And I am happy to take any questions in the panel discussion. But thanks so much for having me, and I will see you soon on Zoom.
All good, everybody can hear me? Okay. So welcome to the invasive mussel session at the Delta Invasive Species Symposium. My name is Marcel. I'm from the Department of Fish and Wildlife, Senior Science Supervisor in the Invasive Species Program, and it's my pleasure to moderate the session. We're going to have 4 presentations with them in person, and we'll also have one recorded presentation, the second one. But before we get to our speakers, I wanted to introduce this topic a little bit. Obviously, the elephant in the room is open mussel, or wide-based mussels, and I thought it's important to introduce this concept a little bit. So this term, invasive freshwater mussels.
Basically, it encompasses the entire complex of the threat that we're facing. So we got 3 mussels in California right now that are a problem, they all have things in common. They're both they're freshwater mussels. They, to some extent, they tolerate brackish water. They all have byssal threads that you see in that red circle, with which they attach, and that makes them able to biofoul surfaces. They're all small, and they also all have a wide range of environmental tolerances. Problems are their high reproductive and growth rates, and their wide environmental tolerance to a wide range of conditions cause them to be very detrimental for an invasive system. Potentially, as intensive filter feeders, they can introduce significant changes in invaded ecosystems, often with undesirable consequences for the system and those depending on it economically or for recreation. And then also this growing in high densities makes them biofoulers on surfaces, on acids, and also on organisms. So we had 2 invasive mussels in California since the mid-2000s, the quagga mussel and the zebra mussel that you see there. They're presented mussels. They came, we imported them with Colorado River water into Southern California's water systems. The good news is that they haven't been spread past the grapevine further north. Actually, I'm talking about the quagga mussel, due to prevention efforts such as controlling watercraft movements and all of that, inspections, etc., outreach. Zebra mussel is only present in 1 reservoir currently in California, also since the mid-2000s, San Justo Reservoir. That reservoir has been closed ever since. They've been contained. So in comes the golden mussel. And it's here. It's in the delta. And it's already impacting us. And the impacts are what we're seeing, like in this photo, in these photos, the impacts are, we haven't seen something like that with quagga and zebra mussels. So we've seen pictures like that from other states, but this is new to California. And it's happening right here in the delta. So you see anything that's in the water in Stockton, for example, these buoys that turn them around, and they're full of mussels. This acoustic Doppler current profiler from the Stockton Deepwater Ship Channel that got pulled in August 2025, full of mussels, and then the underside of a floating dock in O'Neill Forebay, and this picture was taken by one of our scientists. So also full of mussels, and you see them siphoning the siphons out. So anyhow, so here's this big problems. We're already seeing the impacts in the delta. I'm not going to talk about this one much because it's part of Tanya's presentation. Boats in the delta are starting to get encrusted. The mussels are not just on the boats on the outside, they're also getting inside, potentially into the intake systems, the cooling system, etc., causing potential damage. And I just showed this picture already. This instrument was deployed around the time when the mussels were first detected. Hasn't been pulled out of the water since, and that's how it came out. And picture on the left side, and on the right side you can see what it's supposed to look like and what it looked like after cleaning. So impacts are already being felt. Where did these golden mussels come from? So they're native to China and Southeast Asia, certain regions. They initially spread throughout East Asia and from there also to South America, where they spread very rapidly in hydrologically connected systems. And now they're here. Detections to date, you can see their spread, delta pretty much invaded, and then you see these red dots extending all the way down to Southern California. To that though, this spread can really be explained by the flow of water, by basically places that receive delta water as source water. all these places are connected to either the State Water Project or the Sand Water Project, conveying water to Southern California. So the points farthest south at this point are in San Bernardino and Riverside County in the Santa Ana pipeline. The upside to all this is so far we haven't detected any mussels in water bodies that are not hydrologically connected to the delta. And that would potentially be the case through overland movement of both. So this hasn't happened yet, and hopefully it stays that way. So mussels were first detected last October, and the state has responded with convening a task force pretty soon. Multiple agencies, both federal and state, and many partners were joining. And this whole effort kind of culminated in a response strategy that focused on several objectives. The ones that are grayed out are not being currently actively worked on. The ones that are not grayed out are currently being worked on. Communication outreach. Prevention is really one of our main foci, like preventing places where the mussels not are from getting mussels. Monitoring is a big part. So I'm also lead of the monitoring team for the Gold Mussel Task Force. Partner engagement is a big thing, and science and capacity building is part of this partner engagement. So working on shared science priorities, identifying shared priorities and making them happen. And some of the talks that are going to hear today are, so progress has been made, and you'll hear about some of that progress that has been made on some of these shared priorities. Worth mentioning also that regulatory action has been taken pretty quickly. So this Department of Food and Agriculture has rated golden mussel as an A-rated pest. State Lands Commission has put in place emergency ballast water regulations. CDFW has added golden mussels to the restricted species list, and there was this trailer bill that was passed by the governor in September that replaced our regulatory quota language for resented mussels with invasive mussels. So we have the same authorities now for golden mussel that we have for resented mussels. So these are all good developments. And yeah, one of these was also adding a definition for invasive mussels being capable of spreading in freshwater and listed in Title 14, Section 6 on the restricted species list. And this is also forward-looking in case there's other mussels that we have to deal with. And with that, I think I just want to quickly talk about this. So one of the bigger, the foremost priority right now is prevention, and really preventing mussels from spreading elsewhere, spreading in California, spreading across the state line, spreading across the country, across the continent. And what you're seeing here is a graphic that shows where boats. This is from the Watercraft Inspection Database from California, from Colorado. That's maintaining this database, basically showing where boats went in 2024. That from the Delta where they were going. And for example, Lake Berryessa got more than 800 boats that were tracked, and so. You know, of course, you know, that explains why the lake got closed. I mean, it's a big problem. So one of the things we're working on preventing this kind of spread. And yeah, I don't want to get into all this, but really one of the biggest priorities right now is getting a handle on watercraft inspection and decontamination, boosting our capacity in the state, doing more trainings, facilitating others to be able to do that by providing resources as well. And I want to leave it at that. As I mentioned, science and capacity building is a big priority, and some of the talks that we're going to hear are going to be showing you some of the progress that has been made in that area. And the first talk that we're going to have is by Lina Ceballos from the State Lands Commission. And she's also going to be talking about something very important, which is the prevention of spread to other ports of call, amongst other things. And she's also going to tell you a little bit more about the likely sources of the mussels that we're having here, what we know and don't know. So, but before I turn it over to you, one last appeal to all of you. So please help spread the word about this important problem, the concern. If you're in the field, please keep an eye out for invasive mussels, report them to us, and help and promote and implement prevention. Our clean trade dry message, there's records in the back that you can take, share as appropriate. So please help. And with that, I would like to turn it over to Lina.
Thank you, Thomas, for setting up the stage. Good morning, everyone. Thank you for being here. Today, I'm going to talk about the knowns, the unknowns, and assumptions about the role of ballast water in the introduction of golden mussel in California waters. And I'm also going to touch a little bit about a research that we have been working on the potential secondary spread of these golden mussels. So first, let's talk a little bit about background, as the vessels are the main vector for non-indigenous species. We know that shipping is responsible for up to 79% of the established non-indigenous species in North America, and mainly through ballast water and biofouling. So we know that prevention through vector management is the most effective way to reduce the risk of invasive species transfer. This is a very simplistic diagram of the biofouling process. Many of you may be familiar with this, but I'm just gonna go over it really quick. So we start with a vessel in the source port, and the organisms start attaching into the wetted surfaces of this vessel. And then the vessel moves to a new port, a new location, transferring all these organisms attached to it. And then if the conditions in these new locations are convenient and they are optimal for these organisms, they start to detach and spread and spawn, and they could establish and become invasive. Now let's talk about the ballast water process. Again, some of you may be familiar with this, but some of you may not, so I'm just gonna go over it really quick. So when a vessel is in the source port and is discharging the load, the cargo, it needs to uptake ballast to compensate for the weight. So for stability and compensate for weight, so whenever is discharging cargo is uptaking ballast water with whatever is in that water. So, and then the vessel moves to a new location and is doing some cargo operations, loading cargo. And then as the vessel loads the cargo, it is discharged this ballast water with whatever is in the water. So this is a picture of what ballast water would look like if it wasn't managed. So it's full of larvae and all kinds of organisms. But how is ballast water managed or regulated? So we have two main mechanisms to manage ballast water. The first one is ballast water exchange, which, what do we mean by that? Is the ballast water exchange is when the vessel that took water in coastal environments, like ports, with a lot of organisms, it goes to open ocean and it exchanges that water for oceanic water. This oceanic water has a low concentration of organisms, and then reducing like that the risk of transferring non-invasive species into coastal environments, because oceanic organisms are less likely to survive in coastal environments. So the other mechanism to manage ballast water is through a ballast water treatment system. So what is a treatment system? Treatment systems are systems that are installed in vessels. These are technologies that are designed to treat the ballast water. What do we mean by treat? Pretty much killing the organisms that are present in the ballast water with the the objective of obviously reducing the risk of NIS or non-indigenous species transfer. With these two mechanisms are intended to reduce the risk, but none of them actually completely eliminates the risk. So what do we do in California currently? So to manage ballast water, we have adopted what we call the discharge performance standards. And these are basically a number of organisms allowed in a ballast water discharge. So as you can see, it's not zero. So again, it's a risk reduction. However, it's not a completely elimination of the risk. And by the way, these standards are exactly the same as the federal standards. Now let's talk about the golden mussel introduction, what we know and what we don't know. So we know already, Thomas told us about it. We know that it was initially detected in October 2024, and this detection included a lot of multiple sizes. We can infer that the population was really well established there when it was found. What we don't know is the actual introduction date. So we don't know if it was one single introduction or multiple introductions because of the same reason of the sizes. Thomas also touched on this. We know the native range of the golden mussel is here in the map in blue. We also know the introduced range in red, including South America and now most recently California. What we don't know is the unknown introduced range. So if it exists, we don't know if golden mussel has invaded other parts of the planet that we haven't just detected. We also know that the initial detection was near the port of Stockton. What we think we know is that it was likely through a vessel or multiple vessels. And due to the life history of the golden mussels, we think that it could have been through ballast water, maybe as a larvae. Can we really rule out the potential introduction by biofouling? Not really. And most likely we're never gonna know the specific mechanism of introduction. Some of the assumptions that we hear about the golden mussel introduction in California is that this introduction was because of a non-compliant ballast water discharge. And the reality is that compliant ballast water can reduce the severity and the likelihood of transferring or introducing invasive species. However, it does not completely eliminate the risk. Even if we had 100% compliant discharges, we still will have a little bit of risk of introducing invasive species. So another assumption that we have is that we can determine the specific vessel that brought this mussel to California and why we don't go for it. Again, as I mentioned before, it will be very difficult, if not impossible, to go, to point someone if we don't know the specific date of the introduction. So now I'm going to move a little bit to a research that we have been working on in collaboration with the Smithsonian Environmental Research Center. So this is a work in progress, but I'm going to give you a little bit of a teaser for this paper that is coming up. So in this paper, we talk about all the historical patterns and the shipping pressure in California waters. And we're also going to analyze some of the potential spread after now we know that it's already invaded California. So the main three objectives for this paper are evaluate the shipping pressure historically. And we're looking at all the known locations with already established populations of golden mussels that have come to California since the 2000 to 2024. And after we analyze all that shipping pressure and kind of tease apart how and where it came from and all the potential ideas. Then we're gonna move into the potential coast-wise spread. So we wanna know, now that it's here in California, where could it move to? And we're gonna do it coast-wise, Pacific coast, and then also other East Coast and Gulf locations. And this is some preliminary data from this research. So in this graph, we have the number of arrivals from that subset of vessels that comes from already known populations of golden mussels to California, brackish, and low salinity ports since 2005 to 2025. So as you can see, it had varied a lot. Let me see. But we further spread this or divided this to look at the two specific vectors. So in the top graph, we have the total wetted surface area, which is a proxy for biofouling. This means all the area in a vessel that could potentially be colonized by biofouling organisms. And the bottom graph is the volume of ballast water discharge in California throughout time. And this is divided by vessel type. So one of the main things that we can observe in all these graphs is that bulk carriers have been putting a lot of pressure throughout time in our California waters. So I also mentioned that we're analyzing the secondary spread. So for this, we're looking at all the vessels that are coming to our low salinity ports, brackish and low salinity ports, and are leaving, uptaking ballast water and are going to other locations. So in this graph, we show the, sorry, I'm just to point with the laser. So this is a shorter time frame. We're looking from 2019 to now, because this is the time frame where we think golden mussel arrived to California. So we want to know from that time where is potentially going. So in this graph, we see the volume of discharge in the Pacific coast and some in the Gulf and the East Coast. So what are we doing now? Thomas mentioned that we adopted emergency regulations in this year, in the summer. And what these regulations are trying to do is further protect our low salinity ports by creating additional requirements for vessels that are intended to discharge low salinity water in our low salinity ports. So every vessel that is coming with low salinity water and is planning to discharge ballast water in our low salinity ports are now required to do oceanic exchange of the ballast water, plus the use of a treatment system. So it's a double mechanism to protect. Again, why exchange? Because the high salinity ocean water acts as a biocide for all the freshwater and brackish water organisms. And now we are currently during the rulemaking process to make these emergency regulations regular. And we are during the public comment, so I cannot answer questions about that. But hopefully they'll be adopted soon. The last thing that we're doing is we created a system to alert Oregon and Washington for any vessels that are taking up ballast water in Stockton, potentially contaminated with golden mussel, and they have their next port of call any port in Oregon or Washington. So every time that we see a vessel that is taking up water in Stockton and is going to Oregon or Washington, we inform them so they can get in touch with the vessel and make sure that they use proper management before they get there. I think that's it. I will take any questions.
What's the question if we found mussels in the Liberty Island area?
Yeah, wondering if I can triple check.
Okay.
I'm not the one responsible.
Nothing confirmed yet, but bound to be happen sooner or later.
I was just wondering, with the information that you're sending like to Oregon and Washington, are you keeping within that, like how long they were in port?
Well, do I repeat
the question?
Yeah.
Okay, so she's asking if the information that we are sending to Oregon and Washington, what kind of like vessel activities are we like? Good. Okay, so what we're doing is like every vessel that comes to the port of Stockton and does not discharge ballast water, we assume that they are uptaking ballast water. So that's kind of the tag mechanism. If they're not discharging, then we're assuming they're taking water and they're going there. So we just tell them there is a vessel going your way with ballast water from Stockton. And they will get in touch with the vessel and make sure that they have a treatment system that is functioning and that they do the proper exchanges if needed. One more?
Yeah. Can you please hold your question? Thank you, Lina.
Okay.
So, and I apologize, we're going to have a Q&A at the end with all the speakers, so I missed to mention that. And so I want to introduce our next speaker, which is actually a recorded presentation by Tessa Beaver from the Smithsonian Institute. It's a related study, also collaboration between the State Lands Commission and the Smithsonian. And the name of the presentation, the title is Hull Fouling as a Mechanism for the Introduction and Spread of the Invasive Golden Mussel, Limnoperna fortunei, in North America. And Tessa is a biological technician at Smithsonian.
Hi, everybody. My name is Tessa Beaver. I work with the Smithsonian Environmental Research Center in Maryland and the Invasions Lab. And today I'm going to be discussing some recent experiments that we've done with golden mussels, looking at the potential for transoceanic and coastwise dispersal by hull biofouling in North America. And before I get started, I want to give a quick thank you to Greg Ziegler, Whitman Miller, Amy Freestone, and Greg Ruiz for all of their support with this project. So let's get into it. We'll start off with the tiniest bit of background just to kind of get everybody oriented. So the golden mussel, also known as Limnoperna fortunei, is a freshwater mussel that's native to China. So Limnoperna is invasive in other parts of the world, mainly in South America and some other parts of Asia, but it was recently introduced for the first time in North America in October of 2024 in the port of Stockton, which, as many of you probably know, is a major freshwater port in California. So since then, they've been spreading pretty rapidly through the state, presumably enabled by California's aqueduct system and possibly assisted by small boat traffic. You can see just how far they've spread on this detection map that I have here that's provided by California Fish and Wildlife. So one of the first and most obvious questions that we have is, well, how did they get to California? And the answer to this question is likely going to be commercial shipping. So this is definitely the suspected vector for introduction, likely from Asia, based on shipping data and the known distribution of Limnoperna in other ports. Now, there are two main ways in which commercial shipping can bring over unwanted hitchhikers like Limnoperna. The first of these vectors is going to be ballast water. And basically the way that this works is ships will take on large volumes of ballast water to stabilize the vessel in a source port. They'll travel some distance and then they'll discharge that water elsewhere as they load the ship with cargo, and that would be the recipient port. All sorts of living things can survive in ballast water, and in the case of Limnoperna, there could be microscopic larvae that get released in a location that's favorable for their survival, like the port of Stockton. Now, the International Maritime Organization and the U.S. Coast Guard both require that vessels traveling internationally have ballast water treatment systems to prevent this kind of thing from happening. But treatment systems can fail, or vessels might not maintain their systems properly. And this is especially true for the size class in which mussel larvae fall into. They're a much bigger size class than a lot of the other microorganisms that are getting treated, and they're harder to kill. So that's definitely a possibility. Now, the second factor, and the main focus of my talk, is hull fouling. So mussels can easily colonize the underwater surfaces of ships, and they can stay attached throughout a voyage using their byssal threads. Now, if attached animals reproduce and release gametes or larvae in a favorable environment, they can easily become introduced and then invasive. So in the photos that are shown here, you can actually see some golden mussels that were stuck to the bottom of a pontoon boat. I think this was found at Folsom Lake. And thankfully, these were caught before the boat entered the water. But large ocean-going vessels actually don't have the same inspection standards for whole fouling that smaller recreational boats do, at least in the US. And here, whole fouling isn't currently regulated for internationally traveling ships in the same way that ballast water is. So that's an interesting thing to kind of take note of. Now, as we know, Limnoperna is a freshwater mussel. So whole fouling as a vector for their transport is really going to depend on how well they can survive in saltwater, right? So at least in the context of ocean-going vessels. So say you have a vessel sitting in a freshwater port in Asia for some amount of time and Limnoperna attached to the bottom of that boat. The ship leaves port, goes out into the salty Pacific Ocean, and enters another suitable freshwater port. If those mussels are dead by the time they arrive because of all of the exposure to salt water, then introduction's a non-issue. But if they're able to tolerate the increased salinity and end up surviving the trip, then you might have a problem. So only a few studies have examined tolerance to higher salinities in Limnoperna, and this first figure I'm going to show is from Deaton et al. 1989. Now, the units here are a little weird because they're in osmolality instead of salinity, but you can just pay attention to this green box here. So an osmolality of 1000 is about equivalent to what we would see in full strength seawater. And here it's showing that there was 0% survival of these mussels after 1 week spent in this osmolality. And even after they had been exposed for 4 weeks prior to these gradually higher osmolalities. And so this is just showing us that the mussels used in this experiment didn't survive. There's another experiment, though, that I want to talk about. This figure is from a paper that tracked survival over time at 5 different salinities, and the highest salinity they used here was 12 parts per thousand, which is a lot lower than regular seawater. It's about a third of what you would see. And this is showing us that there was 0% survival after 240 hours, but at 96 hours, there were still some surviving. So, and what I really want to get at by showing these two figures is that while these studies were useful in terms of describing Limnoperna's environmental preferences and their survival, we still can't really infer how they would survive on ocean voyages and how much of a problem whole fouling might be for their spread. Also, both of these were ran at higher temperatures than you would expect in ocean water, which could drastically change the results for survival based on their metabolism, right? So that's kind of the previous work that's been done. So for our study, the first thing that we wanted to look at was whether or not whole fouling could have been a vector for the initial introduction of Limnoperna in the port of Stockton. So we found that the closest freshwater port with Limnoperna that also has shipping exchange with the port of Stockton is in Japan. So this is about a 255-hour transit, a little over 10 days for your average container ship. Now, if whole fouling was the vector for introduction, then some proportion of mussels should be able to survive this voyage if we simulate it in the lab, right? So we were also curious to see, though, if there were any other freshwater ports on the West Coast that could be susceptible to invasion by Limnoperna. And in that case, the closest major freshwater port that meets their criteria for survival is in Portland, Oregon. So this is a much shorter transit, only taking about 36 hours. So that leads us to our 2 experimental questions. Our first ones, and these are kind of our big picture questions, right? So the first one is going to be, was whole fouling a possible vector for initial introduction of Limnoperna in North America? And our second question is, what is the potential for whole fouling as a vector for further spread of Limnoperna on the US Pacific coast? So then we have our kind of smaller scale experimental questions that we are using to kind of get at these bigger ideas. So we want to know, can adult Limnoperna survive that 255-hour simulated Pacific transit from Japan to Stockton? And we also want to know, can Limnoperna survive the 36-hour Pacific transit from Benicia, California to Portland, Oregon? And we use Benicia as our source port rather than Stockton because it's a little bit closer to the coast and still meets the requirements for their survival in terms of salinity and temperature and all that. So before we could run these simulations and do any of these experiments, though, we needed to get some mussels and learn how to culture them in the lab. And this definitely took a bit of trial and error before we finally got them happy and healthy. So we started by getting about 400 mussels shipped to us from our contact at California Fish and Wildlife. They range from about 8 to 25 millimeters in length. And then we had to decide on our parameters for culturing them. So you can see here on the right side, this is some of the freshwater algae that we had ready for them when we first got them. So we had to learn how to grow this freshwater algae. We mostly work with saltwater species here, so this is a whole new thing for us to do as well. And so we use these two species, Neochloris and Nanochloropsis, and these were good candidates to start with because they've been cultured before and they're in the correct size range for mussels to be feeding on. And we definitely had to dial in their culture media, learn how to grow them well, and that's a whole other separate thing. So by the time we got the mussels, though, we had all of the algae sorted out so that we could immediately start feeding them. For their culture water, we ended up using coarsely filtered pond water from a pond behind our main lab building. And we put them in these orange buckets you can see here in one of our incubators. We set the temperature to 18 degrees Celsius, which is similar to the temperature at the collection area they came from. And Limnoperna also prefer lower lighting levels, so we put some barriers up in the incubator to dim the lighting a bit. I would also like to add that to our knowledge, this is the first time that golden mussels have been cultured in a laboratory setting. So this was a really big win for us. It was really exciting. From all of the papers that we read about Limnoperna, a lot of the people who are doing experimental manipulation on adults are they have labs in locations where you can just go grab Limnoperna from your local body of water. Given that we are in Maryland and thankfully we don't have Limnoperna invading here, we do have to learn how to culture them and keep them long-term in the lab in order to run any experiments on them. So yeah, that was great. Now we can actually get into the methods for our experiments. So I want to be clear before I start that we ran 2 separate tests for fresh mussels, or with fresh mussels for each simulated voyage. But aside from the saltwater exposure time, the methods were the exact same. So I'm going to present them together in one slide, and you can just imagine this as 2 completely separate experiments. So the first group that we have here is the Pacific Challenge group. Now, these are the mussels that are going through this simulated Pacific Ocean voyage. And we had 10 mussels per bowl with 5 replicate bowls for each group. So each of these bowls was exposed to 32 PSU seawater for either 255 hours or 36 hours, depending on the experiment. And we kept these at 10 degrees Celsius, which was the lowest average ocean temperature that we might expect to see in the temperate Pacific. And this kind of represents a worst case scenario because the mussels would be more likely to survive at lower temperatures due to their slow metabolism. So we also had 2 different control groups. The first of these was kept in normal freshwater, but cooled to 10 degrees to account for any effects that the lowered temperature might have on their survival. And the second group was kept in freshwater and also at their normal culturing temp, but handled the exact same way as the rest of the mussels. So each replicate group of mussels was treated the exact same, and after the exposure portion of the experiment, all of the mussels were moved to individual containers with fresh water for monitoring, and we examined each dish for mortality and or spawning each day. We also fed the mussels daily throughout both experiments, and we provided them measured portions of either saltwater or freshwater algae, depending on the salinity of the water they were in. So during the exposure portion, we provided the Pacific Challenge group mussels with saltwater algae, and we were pretty sure that they would just be closed the whole time, and they were. To our knowledge, they did not feed on that algae, but we wanted to provide it just in case that had anything to do with their survival. We really wanted to simulate what it would be like to be on that voyage. So that's pretty much the methods. It's pretty straightforward. Now we can get into the results for each of these tests that we ran. So we'll talk first about the 255-hour transit from Japan to Stockton. So for our Pacific Challenge mussels, none of them survived. And so this really means that it is pretty unlikely that Limnoperna would survive this voyage as adults attached to a ship. Thankfully, though, for both of our controls, we had 100% survival. So that's going to mean that we had no effect of the lowered temperature on their survival, which is great, and no effect of container switching or handling of the mussels on their survival either, which is also good. We also didn't observe any spawning in this group. So That's our results for that test. Our second test, though, was a little bit different. So for both of our controls, once again, we had 100% survival, which is exactly what we want to see. However, for the Pacific Challenge group, we actually had 70% of the mussels survive that 36-hour transit in salt water. So this means that it is likely that the majority of attached Limnoperna would actually survive this voyage. So that definitely rings some alarm bells for us, right? And the results of these experiments, while they're really simple, and we really just have kind of this one data point of like how many survived, how many didn't, there's huge implications for this, right? And so I'll get into that kind of now. So the first thing that we can kind of glean from this work is that fouling adults were probably not the initial vector for the introduction of Limnoperna into North America. And transport of larvae by ballast water is a lot more likely. And there's actually another reason why that could be more likely too when you think about it. So the fact that this is a 10 to 14 day transit, depending on where you're coming from Asia to the port of Stockton. If you are a ship and you pick up Limnoperna larvae in your ballast water, they have 10 to 14 days to develop in the ballast water and reach settlement stage. So this means that by the time they're getting discharged into the port of Stockton, they are probably old enough to immediately attach to something, and this would prevent them from getting washed out with tidal currents and where they would get washed out to saltier water where they wouldn't survive. So it does seem like ballast water is the much more likely vector for introduction here. The second thing we learned from this is that there is a really significant risk of Limnoperna spread to other freshwater ports, specifically in this case, Columbia River and Portland, by hull fouling. So we know that preventing further inland spread is really important and that AIS checkpoints for recreational boating are critical. But it would also be wise to monitor the fouling of commercial vessels as well, and specifically those with nearby freshwater destinations, because we know that Limnoperna can survive that transit and could get spread to those other ports. And so that's kind of what we learned here. It's really a couple of simple experiments with not a lot of data, but you get a lot out of it. So that's really great information for us to have. And for our lab, our next steps are definitely looking at getting some more mussels, running some more voyage simulations, and really understanding more about their reproductive triggers. So we want to know kind of what the upper limit is of their survival, and we also want to know what causes them to reproduce, because that's ultimately going to be what makes the difference between them just being able to survive and get to a port and them being able to actually be introduced and become invasive there. So that's all I have for you today. I really appreciate you all taking the time to listen to my pre-recorded presentation here, but I will be online and ready to answer any questions that you have. So thank you so much.
And we're gonna move on to our next speaker. So our next speaker is Jonas Grantman from the Corps of Engineers National Aquatic Plant Control Program. He's a technical lead, and he'll talk about a station placement optimization tool called Spot.
All right. Thank you, Thomas. Thank you, everybody. Appreciate the opportunity to come out and talk. I think this guy works. Yeah, so we've affectionately named this tool Spot. I think we had a couple names floating around as we were in development, and I've just, for this presentation, decided I'm going to choose, and this is what it's going to be. So it's official now. All right. Cool. So I just want to get my acknowledgements and thanks out of the way. We have a great team of people we've been working with on this, not only in development, our research partner, our contractor, Motif. And I do want to point out Forrest, who's going to be able to demo the product for us later. So over the lunch break or later in the day, if you want to find him, he's got the live tool up and running. I didn't want to try it up here because that always goes poorly. Some other individuals from there. We have some non-federal partners that we've been kind of testing this with back and forth to try and find the questions that they want answered and how we can develop it better. And then obviously we have a lot of core staff out here in California that were kind enough to invite us in from the national program to help them with some issues over at New Hogan and also Lake Sonoma. So just a lot of thanks for everybody. And this is just how I feel. I work with a lot of really, really smart people. And so a lot of times I'm just kind of at the table like, yeah, that sounds good. All right. This is really, really small for me. Okay. No, so why are we involved as the Corps of Engineers? I assume everybody knows us as an agency, but maybe not. So we're a water resource agency. We have infrastructure, recreation, lakes, flood control, navigation. Water resource agency, aquatic invasive species obviously impact every single component of our mission. So we are very interested in AIS and aquatic invasive species. We work everywhere, despite what this map shows. This is like a sub-program, but we're nationwide. We work in the territories as well. And how I am particularly involved and how we kind of got kicked off with this is through our watercraft station cost share program. So we support state efforts in these highlighted regions with watercraft inspections, helping fund those, helping bolster their efforts so they can do more, increase hours, having better resources on the ground. And this is an older map, so I don't have all of the dots, but the red dots are, stations that we directly support. We have quite a few in also Washington and Oregon. They just fell off this map because I forgot to include them. Anything else here? Yeah, and we do, I will point out, we do partner with, this map is trying to tell a couple different stories at once, but I will point out that we do work with in the Russian River Basin as well. That's one of our authorized regions for this particular cost share program. So we actually just yesterday signed our final agreement with Sonoma Water to help fund inspection stations in that region. But we do have authority nationwide. It's just that our funding is often tied to that authority in those specific locations. Okay, so why do we focus on voter movement? I think it's pretty obvious to everybody in the room. If you're here, we know that recreational water traffic are one of the primary vectors to move aquatic invasive species, whether that's plants or invasive mussels, from one water body to another. I like that term, like secondary spread, right? We get the initial infestation via the ballast, and then the secondary spread this way. And this is actually a snapshot from the tool we're developing, showing kind of a roadway network of how watercraft are hauled, between water bodies. And then one reason we wanted to develop this tool in particular is because as we have these state partners who are running these programs, we have an obligation as the federal government, who is providing 65% of that funding, to ensure that it's being used appropriately. We're putting stations where they have the highest likelihood to prevent that spread. And just helping giving them a tool as well to analyze where they want to put the stations, because they obviously have resources and limited resources as well, and want to work within their constraints and do the best work they can. So why are we going to use cell location data, right? I want to point out, I don't see this as a replacement for the individual surveys that we're doing at watercraft inspection stations. We saw that map that Thomas put up, that shows all the different dots where people were coming from out of the Delta. I think this is another tool for the toolbox that helps get refinements on what we're looking at. So it's going to provide additional information on timing and location and things like that. So it's not new, right? We all have cell phones, right? We have all these free applications that we love, and the way they make money is to aggregate that data and location information about us so that they can provide better marketing at us, targeted marketing, use that information to glean more about how we behave so that they can then offer us more products and services. So it's used all around for private sector, public sector, and then I see a lot of use cases for AIS management as well. But anyway, so it's low cost. It's really easily accessible. We were very surprised when we shopped this around to see what it would cost to acquire all of this data. And it's like billions of data points. Am I in the billions? In the billions. And it was actually very surprisingly cheap to get all of this information. It's anonymous, right? We're not tracking cell phone numbers. It gets a unique ID. It's not associated with anybody in particular. So it's all anonymized that way. It's easily updated. You can grab this and refresh it as often as you want, right? I don't know how often they republish the data from the vendors, but it's fairly frequently, and you can just go back and acquire it and update based on your needs. Yeah, and I think another really good aspect of it, it's scalable, and it's also you can use a lot of our geospatial tools on top of it. And so that's some of our next phases in this project, I think. But really quick, what it isn't, right? It's not every single entity who is in that region, right? It depends on the vendor you're getting, what kind of information they're getting, whose app they're buying data from. So it's a representative sample of the population that you're looking at. It's not every single device. It's not every single user. We're not tracking individuals. Let's see here. And it's something that it's raw data. It's just points. This device was here at this time. This device was here at this time. This device was here at this time. You have to do a lot of processing and careful analytics to make sure that you are capturing watercraft, things that you can associate with watercraft, right? We don't know that this represents a boat. We know that this was in a geofenced region of a water body, and we are making some assumptions that this is associated with boat, and therefore the movement of the device is following the likely traffic of watercraft. Let's see here. Yeah, and like I said, very basic approach here. It's billions of data points. It is so much. I think we're still operating on a cloud-based, analytical software. It's not something you're bringing in at this size to your home PC or your work computer to then process this data. We're working with cloud services like Amazon, cloud servicing processing to help process that and get it all lined up. Got to get a lot of water body data as well. What database do we use for the water body stuff? Is it the national?
It's NHD.
NHD, yeah. So that's giving us where all the water bodies are. We're also adding information from states and others on which waters are infested so that we can have that flagged and you can see where things are moving that way. And then, those geofenced water bodies help us show that that device was in there. And then, we just work through a process of asking questions based on our non-federal sponsors, what they would want to see, refine that, figure out where the gaps are, where we're falling down, where the analytics aren't showing us what we thought they would, and then find out ways to truth that out and get it a little bit tighter. Here's an early version. We were actually working on this tool at the same time that the golden mussel population was found here. And so we kind of had our contractor pivot really quick from working on some of our Western state partners and some of their questions, and had them do a real quick look and say, okay, let's draw some polygons around the delta, and then let's see where water traffic, where that's moving outside of the delta, and see where our highest risk areas, based on this data, looks like it's going. And one thing we found out that we were really worried about is that first lake up there, New Hogan, based on this data. And again, it's just another tool. I know we saw the other data from the regional database system, showed it going to a Corps of Engineers facility. So that's obviously very scary for us that a lot of traffic is moving to one of our water bodies. And so we were early on, we were doing in this kind of alpha version, one-off request. We were talking to a non-federal sponsor. They were saying, I'd like to see this kind of analysis. We would go back, we would work it, come back with a little report that said, oh yeah, this is the timing where you'd want to station, on this highway, or here's how people are bypassing your station, potentially, right? They're coming down the interstate, you have one on the roadway. But, a lot of traffic's moving off and taking the side highway, maybe deliberately, maybe it's just a nicer way to drive. So you might want to look at putting it here where you're going to get that pinch point again. But, it was just very static. It was one-off analysis. And we were quickly realizing that the amount of requests that we were going to get from our non-federals as we started talking about this project was going to outweigh our capability to kind of do that on a one-off basis. So we started looking at how could we build a web application or some kind of tool that people could get into themselves, ask those questions, and figure those things out. And so we came up with Spot. Our goal eventually here is an open access application that anybody can get into. AIS managers, researchers, probably the general public at some point. We want to be able to have customizable outputs. So you can take all of this nationwide data and filter it to your state, filter it to the lake of interest, draw a polygon around it, choose 2 lakes to see how things move in and out of those 2 water bodies and infested. And maybe you're the lake manager over here and you want to see how things move that direction, give you the capability to refine at any scale that you would like. Date ranges, time ranges, all that kind of information. And then also, and I don't think we have this up and running yet, but we do also have plans to do some type of output report. So you can get this information, filter it the way you want it, hit click, get not only your maps, but also get some time graphs of travel, some additional information, how many watercraft are going to each thing. And so we should be on that approach pretty soon. Here. Just a couple of examples. This is just leaving Lake Tahoe. We left it's just going anywhere from Lake Tahoe. And we were like, hey, what if we want to see where traffic moves in the afternoon? We know we get a big, this is what we're curious about between 3 o'clock and 5 o'clock. This is your movement pattern. I think it's selected to all days and the entire data set. And then you could refine it and say, look, I want to see what's leaving Lake Tahoe and what's going to Folsom Lake within the last 7 days, right? Because if a boat is out of the water for 45 days, it's a less concerning vector than one that has left a water body and is going to another water body in that, that drying period where it's still potentially viable mussels or viable veligers. And so you can kind of trim it down to show just the 7 days to and from specific lakes. We've also, let me see. Okay, future, the current tool right now. So what's up and live now, we've added more base layers. So if you aren't a huge fan of the dark background and the blue, you can add all kinds of different, base layers for your map product. We got a bunch of different color presets in there. And we do have a problem where if you're looking at 2 very discrete points and there's not a lot of data supporting the network analysis for that, you can bolden that up a little bit so it shows better on the map than what we've got up here. And then we're also adding some username and password functionality. This isn't to gatekeep the data that we have. We want the username password experience so that we can know who's using it, how they're using it, and then we can build that into the justifications for continued funding, to make improvements, to figure out how it's being used so that we can then, support it, justify it, and then make it better. Current status, like I said, what I'm calling our 2.0, I don't know if anybody else is calling that. I just made that up as I was making the slide, but it's up and running. It's currently out for just limited use to some of our testing AIS coordinators, and they're using it and giving us feedback on that. But I think we're going to have it up and running and widely available, hopefully in the very near future. I'm thinking, shake your head, yes or no, weeks, months, I don't know where we're at. Okay, he said yes. So hopefully in just a couple weeks, we should have this available. I do realize I didn't put the link up, but that link may change, so we'll get it out to everybody in some capacity. Next phases, like I said, that user account password, refresh the data, fix random bugs that we're picking up. Get the report open and get it out for everybody's use. And then we see a lot of opportunity here long-term to get additional GIS data sets incorporated, HUC unit, environmental risk analysis. So, this area up here has the water chemistry that is very, susceptible to infestations. And so you can overlay these 2 things and see yes, I definitely want a station here because it's protecting everything back here. And we know that there's a lot of infrastructure and that it's all very susceptible to establishment. So lots of opportunity to add different things. I see great opportunity for rapid response exercises as well. Thank you. And that will be, so a new infestation, if you have limited resources to monitor where your next step may be in the invasion process, you can draw the polygon around that lake, you can select it and see where traffic moves out from there so you can put your monitoring resources at the best spot. And that's everything. So I think I was right on time. I got the 1-minute warning. That was awesome. And I'm looking forward to the Q&A session. That's honestly my favorite part of presenting. So I hope you guys have questions for that part.
Thank you, Jonathan. Okay, that brings us to our last speaker in this session, which is Tanya Belthusen from the Department of Water Resources. And Tanya is an environmental program manager with the California Department of Water Resources in the Division of Operations and Maintenance. She's been working on invasive species for more than 25 years, including with quagga mussels in the State Water Project. And she's leading DWR's Golden Mussel response, including the development of biofouling mitigation measures to protect water conveyance and power generation infrastructure in the State Water Project. And Tanya will talk about golden mussel mitigation in the State Water Project. Thank you, Tanya.
Thank you, Tomas. So today I wanted to just give a snapshot of what we're doing in the State Water Project at the Department of Water Resources to develop and then implement mitigation measures to protect the State Water Project from golden mussels. For those of you unfamiliar with the State Water Project, it is a state-owned, multi-purpose water storage and delivery project that conveys over 2 million acre feet per year and provides water to 27 million people and over 750 acres of farmland. We have storage reservoirs in the Upper Feather River and Lake Oroville, and then we have the point of diversion in the South Delta to the California Aqueduct. Along that route of delivering water, we have a series of the storage facilities, pumping plants, hydro plants, fish collection facility, and up below the Lake Oroville, we have a salmon hatchery. Many of these structures are vulnerable to mussel colonization. And looking at the current distribution of golden mussels, we already saw some maps on this, right, where we know it's in the Delta, and then we have detected golden mussels in the State Water Project along the course of the California Aqueduct to almost to our terminal reservoirs in the South Bay Aqueduct that delivers water to East Bay, the Central Coast. We have the coastal aqueduct there that also we've detected golden mussels. And then more southward in our West Branch to Pyramid Lake, which is right on the Grapevine, and then on our East Branch Aqueduct to Silverwood Lake and Santa Ana Pipeline. So part of our program is detection monitoring for mussels, in addition to routine plankton tows to look for mussel veligers. We also have staff that look for mussels in the course of their work. And so we have found golden mussels on the buoys, on fish screens, in discharge lines during pump inspections, and some of our gate structures. So this are just examples of where we find mussels. And so based on the impacts that we see at ground zero in Port of Stockton area, we're seeing the heavy colonization there. And of course, this is considerable concern for the State Water Project that has numerous pumping plants, as I mentioned earlier. And we are experiencing impacts on our in-water instrumentation. Tomas already talked about this in-water device near the Port of Stockton, heavy colonization. We're also seeing impacts on other in-water instrumentation, such as our water quality sondes, so inline flow meters, and level floats. At Skinner Fish Protection Facility, which is located just downstream of Clifton Court Forebay and at the start of the California Aqueduct, we are seeing heavy biofouling of the mussels on our fish screens or our louvers. Historically, the louvers were raised out of the water and power washed on a monthly basis. With the onset of mussel settlement, that frequency increased to weekly, but it is really not effective. So even without power washing, we're still seeing heavy colonization on the fish screens, which of course affects the function of those screens. The crews are now hand scraping. The long-term mitigation is that we purchased 20 louvers, and there's about 70 total. And so we can swap out the louvers on a routine basis, clean them off on land, and then put them back in. But this will be an ongoing heavy maintenance burden for the staff there. We're also seeing impacts downstream. For example, at Warren Power Plant, which is just upstream of Pyramid Lake on the Grapevine, we're seeing mussel shells in our unit coolers and sump wells. So of course, this affects the function of the power plants and hydro plants. So in response to the golden mussel detection, we rapidly formed a strike team. And this is a multidisciplinary team with environmental scientists, engineers, electricians, the plant operators and mechanics. We have a great team of consultants and contractors that assist us. And we're developing both near-term and long-term mitigation and response plans. So for our near-term, and this is just, the first few years, our focus is on facilities that are, immediately downstream of the heavy infestation area. So that's downstream of the Delta and O'Neill Forebay. And we are trying to implement protective measures for that small diameter piping the embedded piping, such as unit cooling water, service water, and fire water that's in our pumping plants and hydro plants. And then we also have Skinner Fish Facility that is being impacted, and so that's up first as well. Some of the mitigation measures that we are developing and are in the design and implementation phase is medium pressure UV systems, chemical injection, hot water treatment, and development of anything else that we can think of. So for the UV system, it's a hydro-optic disinfection ultraviolet technology. So it's a medium pressure light, and as water passes through, it kills the veliger stage. And so anything downstream of the UV unit will be protected from mussel colonization. And it's a continuous treatment, so that will protect those small diameter pipelines continuously year-round. This technology is in use by USBR at Hoover, Davis, and Parker dams for protection of golden mussels, and has been tested on quagga mussels, and then has been tested on golden mussels in Brazil. It does have some limitations with the 14-inch maximum pipeline diameter, so it's not for use to treat the entire volume of water that we move in the state water project. It's just limited to that critical small diameter piping. This is an example of the installation at a facility in Alabama. We do have challenges in many of our pumping plants, is that they were never developed with this equipment in mind. So we're really trying to shoehorn in a new device. We don't have a lot of available real estate. So it's been a design challenge for our engineers. And the example of a setup, so this is a cooling line intake at Banks Pumping Plant, and behind that white wall is the forebay, and we have the green pipe that comes in, goes through a pump and filter, and then the UV unit would be installed downstream. And as I mentioned, everything downstream of the UV unit will be protected, but that means we still have several feet of pipeline upstream that is not protected. And so we'll be installing a chemical injection port in that location. And moving on, we do have some challenging locations where we have pipelines that are embedded or run underneath the pumping plants until they daylight. And this is an example of one of our pumping plants. That's the red line is the intake of a cooling line that comes in right behind a trash rack, runs under the facility for it daylights. And so we have, in this case, 110 feet of unprotected pipeline. So we need a different method here because UV is not going to work. And so that prompts us to develop other options. And one thing that we're looking at is chemical treatment or hot water treatment of mussels in these sections of pipeline and other structures in the State Water Project. We heard about bringing mussels to your lab at the Smithsonian. Well, we have mussels, so we brought a lab to the mussels themselves. So we have a toy hauler that's been outfitted with laboratory equipment, and it is stationed outside Skinner Fish Facility. And the benefit of this system is that we have a ready source of veligers and adult mussels, and we pipe the water from the intake channel into the lab through a series of bio boxes. They're just coolers that became aquarium. It's nothing fancy. We have a control set up on the top, and then we can run different treatments down below. So in this example, we have the control up top in the blue, and we have a chlorine array and a copper array, and we can change out those chemicals and concentrations as needed. We also look at the life history of the mussel. So when do they spawn? When do we see settlement? And looking at growth rates as well. So some of the work that we've been doing is on copper exposure. So this is nothing new. We are just taking what's known for quagga and zebra mussels and applying it to golden mussels, trying to calibrate the dosage and contact time. And some of the copper pesticides that we've been looking at is EarthTech QZ and Kutrine Plus, looking at concentrations ranging from 100 to 500 parts per billion. What we are finding is that golden mussels require higher concentrations and longer contact time compared to quagga and zebra mussels. In this graph, the red and green dots are the concentration and duration for quagga and zebra. The golden dots are for the golden mussels. So higher concentrations, higher contact time. We did find that continuous low dose copper works to prevent settlement. And we are finding that smaller mussels are less resilient than larger mussels. So we can kill the smaller mussels faster than the larger adults. And then we also found that adult mussels in post-spawn body condition are not as hardy as pre-spawn. So we're finding that we can kill them in 2 weeks compared to 30 days, depending on that life stage and body condition. Some other work that we've been doing is doing really low dose chlorine and copper exposure trials, and this is to prevent mussel settlement. It's a known method for quagga and zebra mussels, and we have confirmed it does work on golden mussels. It doesn't necessarily kill the adult stage, but does prevent settlement. And then we're also looking at potash, which is kind of a fish-friendly method. So we're exposing adult mussels to potassium chloride, looking at 100 to 150 milligrams per liter for these first trials. There is a national effort to get potash registered as a molluscicide. Canada has gone through the process as well. And then we'll need to work on registration in California. But this could be a viable method in areas where we have sensitive fish species or other species. We've also been doing some testing on hot water exposure, looking at the temperatures and duration to kill adult mussels. So we're immersing them in hot water, recording how long it takes to kill them. And this has been a validation of the current methods that we use to decontaminate boats with hot water. For the decontamination methods, there's 140 degrees for 10 seconds, and we're finding something similar for the golden mussel. So we do know that our current decon methods do work for goldens. So looking beyond, we're going to continue refining our chemical treatment methods and looking at seasonality and body condition of the mussels. We're still in the design phase of the UV unit, really close to 100% on some of those. And then over time, that's just for the first 5 pumping plants in the State Water Project. We still have 30 more to go. So huge undertaking. We definitely have job security. And then we have a lot of other structures that we still need to address, which is turnouts, trash racks, or traveling screens to control aquatic weeds. All of these things are vulnerable to mussel colonization. Also, our water quality stations that continuously draw in water through small diameter piping. All of it is potentially impacted by mussels. Lastly, we have some resources out there. We have a website with what we're doing in the State Water Project with golden mussels. We've also put out some social media videos that are available if you want to see the history of the mussel in the State Water Project, along with some outreach materials. Thank you.
All right, awesome. Welcome, Lightning Talk speakers. Our first speaker is Sky Jung from University of California, Davis, talking about first observation and spread of the Oriental river prawn, a new invasive shrimp in the Sacramento-San Joaquin Delta.
All right, so welcome back, everyone. And today I'm going to be talking about the first observation and spread of the Oriental river prawn in the Sacramento-San Joaquin Delta. I'm Sky. I'm currently a grad student over at UC Davis Center for Watershed Sciences, but most of this work was done while I was at CDFW as a site for the San Francisco Bay study. While I was working there, I was paid by USBR and Department of Water Resources, and this project wouldn't have been possible without the help of the U.S. Fish and Wildlife Service, a bunch of IEP projects, and of course, University of California, Davis. So Let's begin. So quick species profile, Oriental river prawn. It's a Palomona shrimp, so in the same family as Siberian prawn and Oriental shrimp. I know, a little confusing. The genus is Macrobrachium, meaning long arm. This species name is nipponense, referring to their native range, which is in East Asia. They're a freshwater to low bracket shrimp. They're commercially important because they get pretty decent size. I don't know if y'all can see, they get like maybe that big. And they get their name Macrobrachium because they have these elongated second walking legs, which have claws on them. They're sexually dimorphic, with the males having longer claws, and they're kind of generalist omnivores that eat whatever they can shove into their mouths, which can include everything from detritus to invertebrates to even small fish. So a little bit of introduction history. The first observation was made in 2020 in the Clifton Court forebay by USBR. A couple of years later, in 2022, they showed up in the West Delta in Dutch Slough. This observation was made by UC Davis, and they also showed up in an agricultural ditch in Stanislaus County. And in 2023, a bunch of them showed up in the South Delta in various IEP surveys, and they also showed up in an agricultural ditch in Merced County. So you can kind of see they're following water movement. And in 2024, they showed up in the port of Stockton. This observation was made on iNaturalist. And in 2025, the observations kind of exploded. They showed up in Southern California, in Pyramid Lake, in Silverwood Lake, indicating that they're writing the State Water Project down. These observations were made on iNaturalist. They showed up in a random pond in South Bay. And most recently, they showed up in the North Delta in Ulatis Creek. So why might you have not heard about these shrimp? So big thing is that there's a lot of sampling limitations with shrimp in the Delta, not a lot of projects that actively target shrimp. And when shrimp are caught, they usually aren't reported to species because they aren't really a species of interest. And so that leads to a big data gap in what shrimp we're catching and where we're catching them. And because of that, a lot of people don't really know how to identify shrimp, which is the next thing we're going to be going over. So here is a shrimp drawing of a different shrimp. This is Palomon macrodactylus. And here you can see I've highlighted a couple of things like the carapace, the rostrum, and you can see the second period pod is in red. That's indicate that you do not want to pay attention to that, and I'll explain that later. So here's a close-up of the carapace and rostrum of just kind of a generalized shrimp. Here you can see that I've highlighted a couple things on there. Most important is the hepatic spine, which is behind the orbit, and the branchiostegal spine, which is on the anteroventral edge of the carapace. So here's a new friend. We went over kind of their life history and where they're found, and you can see here that the branchiostegal spine is absent from the anteroventral edge of the carapace, whereas the hepatic spine is present behind the orbit. And another shrimp that they're often confused for, the Oriental shrimp, Palomon macrodactylus. Oh God. What matters is that here there is a branchiostegal spine and there is not a hepatic spine. And let me reiterate that this is the most important characteristic. You can see the difference in the spine arrangements between the two species. Here's a chart showing like what's most important for identification. Again, do not rely on the enlarged claws because both of these species can have those enlarged claws. So potential impacts, they're already present in restorations. This one was caught in Dutch Slough. We don't know exactly what they're eating, so we don't know what their food web impacts could be. And also, they can spread upstream because this guy was found in Ulatis Creek, I believe, last week or 2 weeks ago, so they're not entirely dependent on downstream transport or tidal transport. So last couple things, hopefully I still have time, is we need to start tracking their distribution. It'd be nice to predict where they might be spreading. Get scientists trained up on identifying these shrimp, and also rely on citizen science to fill in those sampling gaps. Cool.
While you take questions, I'm going to work on that ding. Time for questions.
So the question was that they get a lot of shrimp kind of everywhere, and wondering where you can record the observations, right?
Yeah.
This is kind of a side project for me. I'm not really working on this for my PhD anymore, but you could probably upload this to iNaturalist and I could provide input there.
Relative to reporting non-natives in the Delta, Marketable California Department of Fish and Wildlife's Invasive Species Program, and we would recommend you could report those to the USGS Non-Indigenous Aquatic Species Database. They compile reports across all aquatic taxa. And feel free to reach out to the Department's Invasive Species Program as well.
Yep. Okay. So in case you see any more invasive species, feel free to report them to CDFW's Invasive Species Program, right? And then also to USGS. I believe I haven't been in the loop with Bay Study, but I believe they tried reporting it to USGS, but that was during the government shutdown, so that kind of went nowhere.
I was wondering if you could speculate on some of the ecological impacts, if you have any thoughts on it, what they might be doing in the ecosystem?
Yeah, sure. So the question was, what if I could speculate on the ecological impacts of these shrimp? So Palmon shrimp are known fish egg predators, so that could be of interest for a couple species of special concern in the Delta. And just from personal observation, there's not a lot of literature on what their ecology is, mostly on just like what they're eating in aquaculture. But I've seen that these guys are pretty darn vicious. They're extremely cannibalistic, and I've seen 3 of them bully a red swamp crayfish so bad that it basically crawled out of the tank. And yeah, they're evil. They're evil shrimp. So the question was, has there been any control mechanisms? As for control on our part, the only thing I can maybe think of is like fisheries maybe, because they're working on developing fisheries in parts of Eastern Europe where they're also introduced. But beyond that, I'm not sure how else, because these shrimp are also often associated with vegetation, and it's kind of hard to, target them. Oh, and then also we have native shrimp that might get caught in the crossfire. One more.
Do you think there's potential of them, based on understanding their behavior, outcompeting or getting aggressive with the established non-native shrimp in the Delta?
So the question was, could you see these guys competing with and outcompeting the other established non-natives? And potentially, though, there's been some work with these shrimp and another shrimp that's non-native to the Delta, Siberian prawn, Palmon modestus. And what they found is that they pretty effectively niche partition, with these guys taking more of a benthic niche, whereas the Siberian prawn are more pelagic. And this is in their native range where they overlap.
Can you provide a specific location for the 2025 South Bay observation?
Can you provide specific information on the South Bay location? It is on iNaturalist. I don't remember the exact locality, but it's the iNaturalist pin puts it in a pond, it looks like. Yeah.
Awesome, thank you. Okay, so our next speaker is Kenji Soto from California Department of Fish and Wildlife, speaking about a new invasion of a crab species I'm not going to attempt to say.
All right, thank you. So my name is Kenji Soto. I'm from CDFW San Francisco Bay Study, and today I'm going to be talking to you about Neoeriocheir leptognathus, a new mitten crab, a new invasion. All right, so first we're going to do a brief overview about mitten crabs, or genus Eriocheir. We'll break down the name. Erio means woolly, cheir means hand, so hence woolly hand or mitten crabs. They are opportunistic omnivores. They are native to the estuaries of China, Korea, and Japan. They do have distinct species ranges with some geographic overlap. Initially, they were placed into one genus, but through molecular and morphometric methods, it was determined that there's actually 7 species across 4 genera. In their native range, they're sold as food with the gonads of the Chinese mitten crab, Eriocheir sinensis, being considered a delicacy. And then, of course, we know that we see mitten crabs all across the world. Most famously is the Chinese mitten crab, Eriocheir sinensis, which was first detected in Germany in 1912, and then it spread through Europe, going through Holland, France, Spain, and England. In North America, it was first detected in the San Francisco estuary in 1992, and then had a population boom in 1998, and then the population declined in the late 90s and early 2000s, and basically hasn't been seen since 2004, with a couple observations in 2010 and 2019. There have also been some non-reproducing populations that have appeared in the Great Lakes, Louisiana, and Oregon. There's actually one other observation just a few days ago in Oregon as well. Another mitten crab, Eriocheir japonica, the Japanese mitten crab, has been seen in the Columbia River, but that was just one individual. And then there was a established population in Germany, Holland, and Poland from 2009 to 2015. And then another mitten crab that has invaded another area is the Hepu mitten crab, which has invaded in Iraq and Kuwait in 2005. Now we'll get to the star of the show, Neoeriocheir leptognathus, which was first seen on February 6, 2025. A mature 20-millimeter male was observed in San Pablo Bay, just north of San Rafael at the San Francisco Bay Study Station 323. It was caught in the midwater trawl, and it was in 11 feet of water at a temperature of 10.5 degrees Celsius and a salinity of 13 PSU. It was identified by Greg Jensen from the University of Washington. And to my knowledge, this is the first time that it's been reported out of its native range. A little bit about the morphology of N. leptognathus. Sakai first described it as gentle looking. And if we take a look at the carapace, it does look a lot smoother and not as spiky as some of the other mitten crabs. And specifically, if we take a look at the front margin of N. leptognathus, you see that it is flat and slightly convex without any distinct teeth. This is also, P. formosa also has this flat frontal margin. And then both these mitten crabs both lack a 4th lateral tooth. Another feature you can use to distinguish N. leptognathus from the other mitten crabs is the presence of setae on the chelae or the claws. So all mitten crabs do have setae on their claws, but E. sinensis and E. japonica only have setae on the outer and inner portions of their claws. P. formosa has setae on the outer portion only, and then N. leptognathus has setae only on the inner portion of the claws. So what are some potential invasion impacts of N. leptognathus? First off, it is not catadromous like the other mitten crabs, so it's restricted to higher salinity environments, and most likely will not be migrating up and down throughout the estuary to the more freshwater environments, and it will most likely not be having as big of an impact as E. sinensis, which has been shown to reduce native biodiversity, compete with other crustaceans, and has also been it is also a parasite vector. It has caused a lot of erosions from its burrows, and if you can see here, this cast of one of the burrows, they can be quite intricate. It was estimated that since its first introduction in 1912 in Germany, that has caused 80 million euros worth of damages. And that's it. Thank you. And I would like to just thank Sky for his enthusiasm in IDing species and Greg Jensen for identifying this species too. And I'll take questions.
I have a question. It's I'm curious what your next steps are now that you've identified this new species. If you could repeat the question.
Yeah, so the question was, what are some next steps? I think right now we're just continuing to monitor just to see if we see any other ones. I'm just thankful that this one probably won't have a big of impact as the Sinensis. It's a lot smaller. It probably stays around this size, like 20 millimeters, probably like the largest it gets. There isn't a lot of literature on it. Most of it is just on physical descriptions. So I don't know the ecology of this crab. Yeah. Oh.
So why did the first invasion of a similar species not take? Because we don't hear those stories too often.
So the question was what was the first invasion of mitten crabs?
Yeah, and they went away.
Yeah, so we don't exactly know why the Chinese mitten crab's population declined. One hypothesis is that it first appeared with like an increase in temperature, had to do with like an El Nino, and then possibly cooler waters led to its decline. But yeah, we don't have an exact answer.
So the weather cycles repeat. It didn't come back when the conditions were good.
Yeah, so he's saying that the weather or the climate's been cyclical, and we've had El Ninos in the past, and yeah, I don't know. Yeah, sorry.
Don't jinx it.
Yeah. Scott has a question.
Hello. So obviously I'm up there in San Francisco Bay State. There's pretty extensive trawls of the lower estuary, and this is the only one that we caught, and it's odd that it was in the midwater and during a high flow event. So do you think there could be more in places that we aren't sampling, and what those habitat preferences might be?
So the question is, are there more, and what other habitat preferences? All I can say is that they prefer the higher salinity waters, and they're probably in the, I can only guess, so don't quote me on it, but maybe they're in the San Pablo Bay area and higher salinity waters. Yeah, unfortunately, there's just not a lot of information on their ecology. Maybe they're making smaller burrows and we just haven't seen them.
Great, thank you. Okay, so next we have Jeremy Crossland from the U.S. Army Corps of Engineers to talk to us about AIS research and cost share programs. Thank you.
Thank you. So Jonas talked about SPOT this morning, and that's funded out of this program. The program's called Aquatic Plant Control, but we do all aquatic invasive species at this point. The language was changed in WRDA in 2014 from aquatic plants to aquatic invasive species. We keep the name because it gets confusing to folks when it's been one name for 70 years and then we change the authority. So anyway, this is the authority where we work from. That's not that exciting. What are we doing? We're all talking about invasive species today. And really our program, we try to focus in prevention down on the bottom left. We all know that's the best and the cheapest. But we fund work that impacts every cycle or every portion of the invasive species curve. Something we've already talked about a bunch, watercraft inspection stations. Jonas put up a map. We fund work with, I think, 13 partners at this point to cost share on watercraft inspection stations and monitoring. So we're trying to prevent the movement primarily of quagga zebra mussels, but it's all taxa, right? We're not letting them go if they have Eurasian water milfoil on their boat. But everybody knows generally how this works, and this is the bread and butter of what we're doing day in, day out. Along with the inspection stations, we fund monitoring works, right? So we can try to catch things early and do early detection and rapid response. On the right side of that picture, this goes back to some of our stuff we've been talking about. This is from a research effort where we were modeling how zebra mussels would basically stack up on the fish ladder at McNary Dam. So kind of like Jonas described, it's not intuitive, but this is moving super fast, so we didn't think they could, but they find little niches, and then they basically build their own habitat after that, right? So once they get there, and if you repeatedly expose yourself to them, they're gonna eventually take hold, and then they make a little spot for somebody else to take hold, and so on and so on, and then you end up with a fish ladder of mussels. And along with that, the other thing we do is we work with the non-federal partners that we worked with through the cost share to do rapid response planning and hopefully never take action, but eventually action has to be taken. So we fund both planning for rapid response and rapid responses if they are necessary. Oh, it didn't move. Right, so this is Phragmites in Kansas. This isn't actually what we've been talking about, but we can do rapid response activities with our partners to any aquatic invasive species. It's cost shared in the same manner. It's just we have to have a plan in place and go to work like that. And well, we fund, I don't know what the percentage is. It's more than 80% goes to watercraft inspection and monitoring. But these are other things we do through the program. We had talked earlier about aquatic invasive species research. We roughly spend about $5 million a year on research trying to improve how we deliver herbicide to aquatic invasive plants. I mentioned the coatings technology work we're doing earlier. It's all stuff like that, where we're basically trying to focus on national priorities for the Corps and buy down our maintenance costs of what we're trying to do. And then after we do that research, the second line, flower brush and hydrilla, we're trying to take the technology we developed to the field and teach our projects and partners how to use it so they can reduce their costs. And then the reason it's called Aquatic Plant Control Program is because originally we did this on the bottom, where we did basically long-term maintenance control like water hyacinth, hydrilla. I'm trying to think of other species, but where we cost share with a non-federal partner, and we did invasive plant management to basically protect both our infrastructure and theirs from invasive species. So today that's probably the smallest portion of our program, where prevention and all that stuff is the largest portion, but that's where it started from, and that's really, I'm pointing the wrong direction. That's the nuts and bolts of our program is aquatic plant control, and yeah, 80+% of it goes out to work with non-federal partners to try to tackle issues that impact both us and the non-federal partner, whether that's a state or a county, right? It's the principle is we're trying to buy down risk for everybody from invasive species impacts.
Okay, thank you so much. Next, we have Cynthia Powell from Calflora talking about tracking management of delta weeds using Calflora. Thank you.
Thank you very much. Let's start with the basics. How many of you have used Calflora? A third. Wow. Okay, maybe half. Okay. How many of you might have used Calflora, but you're not totally sure? Maybe. Since we start with Cal, there's mix-ups with like UC Berkeley and CNPS and CalIPSI. So we are an independent nonprofit plant database, which is really useful for the invasive plants in the Delta and perhaps less useful for the other invasives that are not plants. This is, it's interesting for me to be in a symposium where we talk about things other than plants. I'm really enjoying it. Calflora aggregates data from thousands of sources. Some of those sources are people like you. And when I say aggregate, some people get like, what aggregate? What do you even mean by that? So I've been coming up with a list of like compile, assemble, collate, gather, collect. Like we bring in data from lots of sources and put it all in one place for you to find it. That's one of our secret sauces. Magic sauces, I should say. Hopefully not secret. What are these sources? What are these thousands of sources? I don't expect you to make it through this whole list. The two that people are usually the most interested in are the Consortium of California Herbaria and iNaturalist. And then there's, thousands of others in there. And so we get regular refreshes of data from all of these sources so that when you're trying to track invasive weeds in the Delta, water hyacinth, alligator weed, for instance, Okay, let me back up. From this aggregated data set called Calflora, which non-native plants currently grow in the Sacramento River Delta? So there are 350 non-native species currently in the Calflora database in this area, and of those, 120 are Calipsy listed. What does it mean to be Calipsy listed? Like, really bad. Calipsy has a ranking system where they say, what should we be really, really, really worried about? And so of those, 120 are in the Delta. Yikes! What are we gonna do? Well, in order to tackle these things, we first have to know what we're tackling, what we're trying to tackle, and where it is. This is the aggregated data set for alligator weed. And all those blue dots come from those thousands of sources I was just talking about. iNaturalist, specimens, state parks, national forests. We pull them all together here, and also from you. We have an app you can use in the field. Gina uses it all the time. Yay! Gina, I think actually,
No, it moved down.
it moved, oh, she had one of the top reference photos, and now it's below Ron's.
That's Robin Carter. She's in DWR too.
Yes, is she here? No, she's not here yet. Some great photos by some people you might know. And we have a team of volunteers you're welcome to join who are refreshing and upgrading or maybe downgrading. Sometimes if it was Gina's photo that got moved down. Oh goodness, what happened? So changing the photos, we have our bloom wheels, we have the family, the category, lots of links to other resources. And this aggregated data set for all 8,500 species that grow wild in the state of California, including this really, really bad invasive in the Delta, is what we have in the Calflora database. So let's look at the water hyacinth. Mike Ressler. So here's a map of the distribution of the water hyacinth. And you'll notice some of these are just red squares and may or may not have a blue dot in them. That's when we know it's somewhere in that quad. We just don't know exactly where. That probably came from Calipsy or sometimes we get, well, for rare plants, we sometimes see quads because they're obscuring the location. So when we get CMDDB rare plants, then you might not see the exact location. You can always drill down and see who the observer is and contact them and ask for the real location if you'd like it. Okay, so this is how Calflora is a database. We aggregate from lots of sources. We're an independent nonprofit, and we are serving the public in many ways, including helping you track invasive plants in the Delta. Now, the title of my talk, I actually just realized this, is actually tracking management of Delta weeds. So in addition to tracking plants, you can also track your management using Calflora. What did you do? Did you use chemicals? Did you use like some other vicious shrimp to eat the invasive vicious shrimp? What did you do? Goats. So that you can also track in Calflora in addition. So now it's time for me to wrap up. Thank you for supporting Calflora, a 501c3 nonprofit. And here is a QR code that my board always says I should have in every presentation. And some of you already support Calflora, so thank you very much. And I'd like to take questions. If your question was going to be, where do you get this amazing Calflora swag? Gina has some too. On our website under news, you too can order your very own Calflora swag. Next question. Elizabeth has a question.
If I was going to, let's say, a park or a location in the Delta or otherwise, could Calflora tell me what plants I'm likely to see part on it?
The question is, if you're going to some park or open space in California, could Calflora indicate, suggest, tell you what plants you might see, invasive or otherwise? Good question, Elizabeth. Yes, is the answer to that. We have many ways where you can kind of look at where you're headed to, what plants there might be there. One of them is called What Grows Here? And on our website, you can go to What Grows Here and say where you're going to go. You can say, I want to see only the rare plants that are in, or only the natives, or only the non-natives, or only the really bad Calipsy listed ones. And then you can see exactly where they'll be. You can even, if you want to take it to the next level, load it into the phone app. So if you're not a botanist, if you're not really sure what you're looking at, and you don't maybe have access to Seek or to the satellites or any connectivity, you can also follow the dot and be like, okay, I'm looking for this rare plant, the surf thistle, I've never seen it before, and then it'll take you to that dot, and you can hopefully find it that way. Great question, Elizabeth. Oh, one more question. Mm-hmm.
Research the essence of distribution and such for the modeling and it holds that potential accountable.
That's a great question. How can you use Calflora for modeling? Which the answer is yes, you can use. We built some models. You can export the data and create your own models, or you can look at the models that we've built showing potential habitat for plants. And the first part of your question, I think, was related to our app, which is called Observer Pro. And it was, how can you use, wait, what was it exactly? Like AI, was it related to AI? No. Right. How do you use the large data sets in Calflora? Yes, and our data sets, at least for plants in California, are often bigger than INAT, since INAT is one of our data sources. So we have the INAT data in Calflora plus all these other thousands of sources. So yeah, you can export by shapefile, by Excel. We have polygons, so you might want a shapefile if you want to collect the polygons. Then you can do your own research and analyze the data. You can create your own models. We also have potential habitat models already on each of these pages, plant range. If you click on that, it'll show you, well, for an invasive, it's going to be, it'll be kind of depressing, but for other, native plants, it's nice to see what do our models predict the plant range to be. Okay, thank you very much.
Okay, awesome. Thank you. Next, we have Bailey Morrison talking to us about mapping and management of invasive aquatic vegetation in the Delta.
Yeah, I'm going to try and do this in 5 minutes, but I'm here to share about a project that I'm helping co-lead to map invasive aquatic vegetation using satellite imagery. And we're really interested in making sure people are aware of this project going on right now, because anyone who thinks that they'll be able to use these vegetation maps for their work or have it benefit their decision-making capabilities, we'd like you to reach out to us, please. So since almost every year since 2004, there's been airborne imaging spectroscopy data collected, which is the fancy term for hyperspectral imagery. And from that, invasive aquatic vegetation, which you'll hear me say IAV sometimes, classification maps of IAV have been made from this hyperspectral data. These have been a really useful and amazing resource for the state. And more recently, a former PhD student in the lab I work in, Dr. Christiana Ade, along with the Delta Stewardship Council, were interested in seeing if we could do the same type of classification maps for IAV using satellite versus airborne imaging spectroscopy data and have comparable output maps for monitoring efforts. And so from her dissertation work determined that there's absolutely potential, and she developed the prototype. And from that prototype, we were able to secure funding from the NASA Applied Sciences Program, along with continued interest from the Delta Stewardship Council, IEP, CDFW, and the California Department of Water Resources, who are not only financial stakeholders, but end users to this project as well. The main goal of this is to take the prototype and operationalize it, which what we mean by that is to have a continuous, long-term, sustainable mapping effort for invasive aquatic vegetation over the whole Delta. That's the main goal, and the main sub-goal of that is that we will be able to have minimally a monthly map of IAV distributions at the pixel level for every month for all years that Sentinel-2 data has been available, which is 2016. Secondly, we're working on advancing some species distribution models to look at how IAB has responded to certain management actions and decisions in the past, such as the emergency drought barrier, and then use those models to potentially do some forecasting so that our end users can, if they're going to have some action they need to do, what's the effect going to be on IAB if we do this? So yes or no, depending on the output from that. And then since we have all these great end users and stakeholders, we're co-developing, collaborating closely with how people will receive and be able to use the Sentinel-2 IAB maps. We're at a point right now where we have a working model that can spit out monthly Sentinel-2 classification images. And we're working on a survey to start gathering information on how we're going to deliver this to people. So one of the questions is, since we have the airborne data, why are we even bothering with the satellite imagery? The hyperspectral, it's got thousands of spectral bands, which have a lot of information for breaking apart classes through imagery. It is also finer pixel resolution. It's about 2 to 3 meter resolution. And in terms of submerged aquatic vegetation, we get, since it's on a plane, we get to decide when it flies. So we can capture data at low tide, which minimizes issues like submerged aquatic vegetation being classified as water because it's too high over it. But the reason we're interested in the satellite imagery, or at least having this as another option, is because Sentinel-2 specifically over the Delta has a revisit of about 5 to 10 days, and it's free. Doing the airborne is really expensive. There's been years that we weren't even able to fly because of funding. And as multiple people have mentioned already today, funding's a big concern right now. So we'll be able to gap fill. And this is not intended to replace the airborne imagery. This is meant to fill in the time in between each annual snapshot. You can see from the previous figure, there are, at least since 2018, over 70 images with less than 25% cloud cover that we'd be able to make a class map for. This is just an example where we can do some phenology work. There's a lot of other type of monitoring questions we can do if we have a longer time series and going throughout the year. And then this is just a quick example, since we're very early stages and starting to develop how we're going to get this product out to people and make it as useful and beneficial to their decision making. So please, if you think of a way that you either use the airborne or could use the Sentinel-2 maps, please reach out to us. We'd love to hear from you. And since we're just about to start creating the tools, this is the perfect time to join it. So thank you. If you have questions, no. The question that was asked is, how did the Sentinel-2 class maps compare to the accuracies of the airborne imaging spectroscopy ones? So there's two kind of differences. The first is the Sentinel-2, we have a few less classes. It's not quite as specific. The hyperspectral you can get for some plants to species level. We can only do water hyacinth and water primrose with Sentinel-2 at the species level. But in terms of the classes that we are able to do out of both, my latest updated version is almost at the same accuracy as the airborne, which is great. There's still some testing we're doing on the seasonal part. Since the airborne's always been collected in summer and fall, we haven't had winter and spring data. And so we've been starting to do that with the funds that we got for this project. So double checking and making sure it's performing well in those months is what I'm currently working on, and we need more data to do that too. There was one more question. Quick one. Quick question.
Questions from Sean.
Of which one? So the airborne is 2 to 3 meter resolution. Spectrally, you get about, I believe, 1,000-ish spectral bands, spectral wavelengths. And then the Sentinel-2, you get 13 bands. 4 of those in the, which is the visible bands, so RGB, and one of the near infrared's 10 meters. So it's a little bit coarser. And so that's going to be dependent, like channels that are smaller than 10 meters, that's going to be a limit. But a lot of things like water primrose patches, there's tons that are huge. There's water hyacinth patches that are huge. We're also looking at emergent marshes as one of the classes. It's non-invasive, but they're being affected by the invaders. And so we're starting to do stuff where we can see how water primrose is shifting into the terrestrial part of these emergent marshes. So there's a little bit of a scale dependency depending on your needs. But those are the things you can contact us for. We can explain a lot more of that. I only have 5 minutes. Sorry.
Okay, next we have Jessica Vollmer from California Department of Fish and Wildlife to talk to us about nutria eradication. All right. Okay. Hi, everyone. Thanks so much for having me. I'm really excited to talk to you today about nutria. I am the program planning specialist with the Nutria Eradication Program with the California Department of Fish and Wildlife. Right. Get going. All right, so I want to play this quick animation here for you. It's a timeline on where nutria have been detected. Over the last 8 years, this white dot in the center, I hope you can see it, is the initial detection in 2017, and that's in Merced County. So in 2017, we start seeing a smattering of detections in Merced and Stanislaus counties. In 2019, we start moving down to Fresno and moving up the river corridor, that's the Merced River corridor. In 2020, we start moving north into San Joaquin County. 2022, we get first hits in Sacramento County, that very bottom tip there. 2023, Solano County in that Sassoon Marsh area. And 2025, we have our furthest south detection, and that's in Kings County. There at the very bottom. I want to draw your attention to a couple of these hotspots. So Merced County, kind of a larger spread. In Fresno County, that large hotspot there is the Mendota Wildlife Area. It's like the Fresno Slough area. And then, of course, the Sassoon Marsh up at the top. And then those two river corridors as well. That's the Merced, kind of the northern branch there going up into the foothills, and then the San Joaquin River. It's more south. So this map, you can see the similar hotspots. This is total numbers of nutria taken. So I want to kind of talk about the total numbers. So the chart on the side, we've got total numbers of nutria taken by year. We start getting a slow increase, but with the wet winters of 2019, our 2020 numbers start exploding. And then start seeing a decrease in 2021, but with the heavy rains of 23 and 2024. In 2024, we get a huge explosion of record-breaking 1,300 plus nutria taken. But this year, 2025, we have over 2,000 nutria taken from the landscape. So yeah, just a huge increase in numbers this year. So as you all probably know, California is very hydrologically connected. And add to that animals which are highly reproductive and can disperse to a great distance. When early detection and rapid response is key, unfortunately, we had to redirect a majority of our northern operations to surveys and trapping in the Sassoon Marsh during non-waterfowl hunting season, while also directing a portion of our southern operations to Mendota and Fresno Slough area to implement a much more intensive trapping effort to remove the nutria from that core area. So it's important to note that we do have staffing limitations. That means that when we intensify our trapping efforts, it comes at the expense of conducting surveys. And when early detection is really important, we're really in a trade-off scenario right now. So always working on opportunities to increase our resources in critical areas like Mendota and Sassoon Marsh, where due to leaving those areas on the back burner and later detections, we experienced those population explosions. All right, so zooming in on the delta, here we can see that the majority of the locations detected in the Sassoon Marsh weren't discovered until later 2023, 2024. Our main focus in this area is working on gaining access from local landowners, other agencies, using newer methods like scat detector dogs and Judas nutria trials, which I'd love to talk to you more about, but low on time today. This chart really, I wanted to show the spread of nutria throughout the last 8 years, starting with low numbers in 2017 in just those two counties, but gaining counties throughout the years, and then
with, I believe it's 10 counties with Nutria detections so far. And then Solano County, which is in the light green line there. Solano is accounts for about a quarter of all Nutria removed from the landscape this year at about 500. So yeah, I'd be happy to take some questions.
And so when you're saying they're taken, is that you guys taking them or like anybody, sending you a picture that they caught one or what have you?
So the question was the definition of taking, is that just us? Is that anybody? So taken is like removed from the landscape, dispatched. Most of those are our program. We do have some landowners who like find them roadkill, for example. Sometimes we'll get a picture and we'll count those in our statistics. Local landowners might be hunting them, but we don't know if they don't tell us. So those are the ones that we are aware of, yeah.
So my question would be the numbers that you have, I mean, that's more than, it might have more to do with your resources of going out and getting them than it is the actual population size exploding?
Sorry? Oh, so the question was, is that more to do with our program being able to find them basically and know where they are across the landscape? It's really hard to tell. We have to gain access from landowners. A lot of people don't want us on their property, but from what we're aware of, that is kind of the boundary of what we know of, and working in those two main areas right now, the Sassoon Marsh and Mendota Wildlife Area. That's kind of our focus right now, just because those are the largest populations we know of. We're of course, spread throughout the landscape as much as we can be. Oh, one more time.
Sorry.
All right, thank you.
Awesome. Find the speakers later and ask your questions. Okay, so next we have David Hammond from Earth Sciences Lab talking to us about protecting infrastructure from golden mussels.
Hi, everyone. Thanks for having me. Being an invasive species event, we hear a lot of important work from people who monitor, survey, map for invasives, people who predict the spread of invasives, prevent the spread of invasives with boat inspections. I just have 5 minutes, so I'd like to be clear at the risk of being blunt. We work in the space of killing invasives, invasive mollusks. That's zebra mussels, quagga mussels, New Zealand mud snails, Asian clams or corbicula, Chinese mystery snail, Chinese pond mussel, and now the golden mussel. What we have is a liquid ionic copper formulation that is registered by the EPA in all 50 states, as well as in Canada and Australia. What's special about it as compared to conventional copper sulfate is that it is liquid. It's extremely soluble. It won't settle out. Its primary means of removal from the water once the water's been treated with it is biological uptake. And we've formulated it in a way that's very bioavailable. And that's important because it means you can use less. And that in turn is important because it avoids impacts on non-targets. Copper, like in many things, the toxicity is all about the dose. At a very low dose, copper is a micronutrient. We need it. In fact, every living thing needs some copper. But as you get higher doses, it becomes toxic, especially to certain aquatic organisms. And we take advantage of the fact that zebra mussels and quagga mussels are extremely sensitive to copper, as are cyanobacteria. More so than fish and other non-targets. So we play with that range just enough to be toxic to the invasives, zebra, quagga, cyanobacteria, but not toxic to the fish and other non-targets that we care about. Most of what we do is protect infrastructure. That's primarily because there's somebody who is responsible for it. They have a budget. And it's a recurring problem. But we have lots of experience controlling zebra and quagga mussels around the country. We have large systems. We protect small ones, very long pipelines, short pipelines. Really never had a failure or a horror story of any sort. What we also do that might be a little bit more surprising for some people is that we also work in open waters. We have worked in lakes, actually eradicating zebra and quagga mussels from entire lakes. I know that surprises a lot of people, but we have done population management within lakes, people just wanting to protect a marina or a dock, an area, a beach, an area of high value. We also work in rapid responses, meaning trying to wipe out the population when it's first discovered before it can spread to the rest of a large lake. And we even work in fish hatcheries, including trout hatcheries, some of the most sensitive fish in terms of sensitivity to copper. We're dosing directly into a fish hatchery year after year. They wouldn't be using the product if it was killing their fish. So, we have a lot of experience in this range of low dose copper. And I would hope, I haven't heard anything about rapid response for golden mussel, but I would, a couple of weeks ago, I heard about a lake where somebody brought their boat up from the Delta, put it in the lake. Later, people said, hey, have you checked that boat? They pulled it out. It had golden mussels on it. They sent the guy back to the Delta to, do what a little too late, right? I would hope that people would consider a rapid response in an event like that, because it's a narrow window where you could still knock them out before they spread to the rest of the lake. We've been working with golden mussels since 2015 because we were expanding into Brazil. And at that time, the top study, they had pretty much discounted the use of copper because they needed such high doses of copper sulfate in order to achieve mortality, like 8 times the EPA limit of 1 milligram per liter. But once they tried EarthTech QZ, the low dose that I'm talking about, it made it more viable. Just 0.35 milligrams per liter as copper was 100% mortality in 30 days. Not all mussels are the same. Quaggas are more sensitive to copper than zebras. Golden mussels are more resistant than either zebras or quaggas. And some of the leading work on that is DWR that Tanya already covered to some extent. We're going to need to use more for control of golden mussels, but that's true of anything you use against gold, any molluscicide. You're going to need more for golden mussels than you would for zebra or quaggas. I'm going to kind of jump ahead because I'm told I'm already out of time. I'll just mention that I know some people have probably been thinking that they're going to use chlorine. If you're at a drinking water treatment plant, chlorine's available. That I don't think is going to be viable just because of cost. Since COVID, the cost of chlorine has gone up 4x or 5x. And so it's now more expensive to dose 1 part per million chlorine than it is 1 part per million as EarthTech QZ, and 1 part per million chlorine doesn't do anything because there's a background demand. And in fact, to treat delta water, you probably need 7 parts per million chlorine just to start to get a residual. Finally, I'll just mention that we have a bunch of plants, drinking water treatment plants in California that have already been using this product as an algicide, and they will be de facto laboratories for us to see whether their dosing into their pipelines protects that infrastructure. This is an example of the water being distributed from Silverwood Lake through a complex series of pipelines. The ones on the right and the left are being dosed with EarthTech QZ, kind of a preventative manner, and the ones in the center, to my knowledge, are not being dosed with anything except perhaps chlorine. And we'll get to see in the next few months, how well each of those is working. Thanks very much. And we have time for questions. Okay. Good question. Yeah, he's asking, how would you apply this in the Delta? I am not proposing necessarily that you would. It depends really on the circumstance, and it's case by case, right? If you had it, the first reaction when they were discovered last October, the first reaction was, oh my God, you need to nuke them. before, this is a horrible invasive species. You need to knock it out before it spreads everywhere. But it was quickly made clear it's already everywhere, and that was not on the table anymore. But, for, I don't know, a Lake Berryessa or, some other lake, if they get introduced there and they're found on a boat ramp, I would hope that people would consider heavily treating that boat ramp to knock them out before they get to the, whatever damage or cost you're worried about with a small treatment like that is nothing compared to the damage and the cost that you'll be facing once they get established.
All right, thank you so much. Okay, so our last speaker today is Jenny Ketterlin from the U.S. Fish and Wildlife Service, talking to us about an AIS horizon scan. Thank you. Okay, good to go. All right, good afternoon, everyone. I'm Jenny Ketterlin, the Invasive Species Coordinator for Refuges for Fish and Wildlife Service Pacific Southwest region. And this will be an overview of a horizon scan conducted for aquatic invasive species in our region by biologists with the Service's Fish and Aquatic Conservation Division. In terms of invasive species, horizon scanning is a systematic examination of future potential threats to an area, and identifying these potential threats can allow us to take actions to prevent their introduction and spread. Horizon scanning is identified as a foundational component of the Department of Interior's National Early Detection and Rapid Response Framework, and the service's regional horizon scans are designed to be repeated over time and vary in focus depending on regional priorities. The objective of this horizon scan was to identify species of fresh and brackish water at risk of arriving in our region, becoming established, and causing harm. This was achieved through a three-phase process. First, a local group of experts was convened to help develop the scope by providing guidance on which introduction pathways, geographic boundary, and the groups of species to analyze. 19 experts from these organizations here participated in this horizon scan starting in 2021 via virtual meetings, a questionnaire, and feedback on a draft scoping document. The introduction pathways selected were commercial shipments of live organisms, recreational watercraft, and hydrologic connections. The Fish and Wildlife Service Pacific Southwest Regional Boundary was selected as the geographic scope, which includes California, Nevada, and the Klamath Basin in Oregon. And it was chosen because the group felt that any preventative regulations for trade or watercraft movement would be implemented along these political boundaries and not necessarily watershed lines. Freshwater and estuarine mollusks were picked as the highest priority focal taxa group due to the types and severity of impacts already caused by some of the species. 3 carp genera and plants in the genera Ludwigia and Vallisneria were also selected. During the second phase of the project, Fish and Wildlife Service staff developed the initial list of species that might arrive in the Pacific Southwest region by estimating pathway volume of donor regions, conducting climate matching, and verifying existing occurrence data. The donor region list included 40 countries with the highest volume of trade to the region, 2 states with a high amount of boat traffic to the region, and 5 states containing upstream or downstream portions of waterways within the target region. The blue states and countries here are all identified as potential donor areas. A climate matching tool was then used to determine climate similarity with the Pacific Southwest region. Species from the focal taxa were selected from the 23 areas ranked as either high in red or medium in orange for climate matching. The initial species list had 164 species in the focal taxonomic groups that were present in donor jurisdictions, but not already present in the Pacific Southwest. All of the originally prioritized groups were represented except for one of the invasive carp genera. During the final phase of the project, the species in the focal area were screened for potential to be a high risk to the Pacific Southwest region using the service's established method for conducting risk screening called the Ecological Risk Screening Summary. Of the initial list of 164 species, 24 were already in our risk screening database, and then 20 additional species were put through the risk screening process based on 3 factors identified by the expert group during the scoping process: a history of introduction, presence in trade, and presence in North America.
Next, the ecological risk screening summary was used to classify risk of these species based on past invasive behavior, climate similarity, and the credibility and reliability of information available and used for assessment. The risk screening tool identified 6 species that were of high risk. The remainder of the species have an overall classification as uncertain, with 3 of those species having a high history of invasiveness, but low climate match to the Pacific Southwest region. And the rest of these species either had a history of becoming established but no impact information, or no history of established non-native populations. So these are the 6 species that were identified as a high risk to the Pacific Southwest region as part of this horizon scanning process. All 6 species were found to be able to be spread through at least one of the focal pathways, and all 6 have been known to hitchhike in live trade shipments. 5 of the 6, all except for the false mussel, are also found in trade either domestically or internationally. It's recommended that these species be added to watch lists for early detection and educational outreach materials to be developed. Additionally, these species could be considered as candidates for state or local regulations were not already listed after further risk analysis and consideration under specific listing rules. And with that, I'll take any questions, or you can also feel free to email this general email list or take a picture of the QR code to get a copy of the full report.
All right, we're going to get started with the final session of the day. Thanks so much to everyone for sticking around. It's great to see so much retention throughout the day. For those who don't know me, my name is Rachel Wigginton. I'm a senior environmental scientist with the Sacramento-San Joaquin Delta Conservancy, and I'm also a co-lead and the facilitator of the Delta Interagency Invasive Species Coordination Team. So I really appreciate you all taking the time to stick around for the 4th session, which is about new tools and novel techniques, really thinking about our theme of managing and modeling for a future delta. We know that conditions in the delta are changing, they have changed, and they will continue to change. So we'll have a couple talks highlighting some of those different types of ecological and environmental changes that we might expect. And we also know that as we move into the future delta, we're going to need new tools, different approaches that we might not have used in the past that will help us manage this changing system. So that's what we're going to be highlighting during today's talks. Adrienne Smits is a research scientist at the Department of Environmental Science and Policy at the University of California, Davis. And they're going to be discussing drought and aquatic vegetation and how those alter water clarity, and fish assemblages.
All right, thanks for sticking around this afternoon. I'll be talking a bit more about habitat change that's been sort of caused by invasive species, and then also the implications of those changes on, well, both native and non-native fish species in the North Delta. So as I'm sure most of you already know, there's been a lot of alterations, in the Delta and its watershed that have worked to actually increase water clarity here in the Delta. So a lot of the dams, for example, are trapping sediment and reducing supply to the system. And actually, so some of the plants that we've been talking about earlier today, they take advantage of increasing water clarity, they establish, and then they can actually do this feedback where they, slow water velocity, they cause sediment to actually settle out of the water column. So they actually make this clarity change worse. And now add on top of that our sort of intrinsically variable hydroclimate we have here in California. We have extreme precipitation whiplash, which of course may just continue to get more extreme as we continue to change the climate. And that can also have implications for water clarity. We see a lot less sediment supply in those extreme drought years. And alternatively, when we get those really big water years, we can see a return of sediment to the system. And so I'm particularly interested in how some of the interactions between those stressors, so hydroclimate and invasive submersed aquatic vegetation, sorry, I should have explained that, SAV, how that's impacting habitat conditions in the Delta, and I'll focus a lot on water clarity. and what that means for the Delta's fish communities, both native and non-native. And so for the rest of the talk, I'm going to focus on the North Delta, so the tidal slough systems, freshwater in the North Delta, and specifically the Cache-Lindsay Slough Complex. So these are terminal systems, and for a long time they were considered potential refuges for native fishes. They'd maintained higher turbidity than elsewhere in the Delta, and they were a bit less invaded by some of these submersed aquatic plants. And yeah, there were higher abundances of native species. And so I'm going to share some work that was done by my collaborators at UC Davis. They were the ones actually carrying out this monitoring study. And so they have been carrying out this decade-long monthly monitoring for fishes and water quality constituents in these two sloughs. And so it provides this really nice data set for us to look at change through time. And another kind of nice aspect of their study design is that Cache Slough and Lindsay Slough can be compared to each other to understand the sort of the role of some of these drivers. So Cache Slough maintain higher turbidity than Lindsay Slough, and at least at the beginning of our study, had not been as invaded by submersed aquatic vegetation. I'm talking here mostly about Brazilian waterweed. Lindsay Slough, in contrast, already had higher water clarity and was heavily invaded by Brazilian waterweed already in 2013. And so, using that amazing monitoring data set, we could start to ask some questions about, what's driving the habitat change in the system and what does it mean for organisms living there. So the first question was just to disentangle the role that our hydroclimate plays in changing habitat conditions, and then how it interacts with this invasive aquatic plant. So how does that interact to affect water clarity in the system? And then how has those changes affected fish assemblages and abundances? And so I'll kind of go over the broad shifts we've seen over the last decade. As I'm sure you all know, we have seen several severe droughts and some really, really big water years as well. So we got a lot of precipitation whiplash during that decade. And the middle panel that you're looking at is a time series of submersed aquatic vegetation volume. So we're kind of using the bycatch of this plant in water trawl nets as our metric of vegetation density. And, I know that has certain problems, but it is actually kind of a nice metric to track change through time in vegetation. So that middle panel is showing you how Lindsay Slough, which is the magenta color, was already invaded with this aquatic vegetation in 2013, but continued to increase. And Cache Slough caught up after a few years. So it started out pretty low in vegetation and unfortunately then, decided to grow some. And then that bottom row is showing you how water clarity, which we're capturing with Secchi depth, changed correspondingly. So you can see that in both sloughs, Secchi depth started out pretty low, about 30 centimeters, and unfortunately, by about 2020, it had increased to about 150 centimeters. And so I wanted to try to understand what the driver variables were for these water clarity changes. And I did some time series modeling of the Secchi depth data itself, and I won't get into the modeling details. I'll just kind of share the highlight results. So those time series up top are showing you the raw Secchi depth data for all 14 of those stations in Cache Slough and Lindsay Slough. And we modeled that data as a function of a few different potential drivers. And we found the two that were the most significant in predicting water clarity were actually local precipitation. So rather than net delta outflow, for example, it was really precipitation within that local area. So when we have those droughts with low rainfall, we see corresponding increases in water clarity. And the effect of vegetation was a little more subtle and kind of interesting. So we found that actually in Lindsay Slough, where it had been already invaded from the beginning of the study. Clarity dynamics were not responding to changes in vegetation. It had kind of crossed some threshold. Whereas in cash slough, which was actively invaded during that decade, we saw the sites that had the lowest initial densities actually show responses to that vegetation expanding. So it looks to me a bit like a threshold effect on water clarity. Okay, so we see these pretty dramatic differences in the habitat within these sloughs. What effect is that having on the fishes that live there? For the sake of simplicity, I'll talk a lot about groups of fishes according to their habitat preferences rather than species. So we have our pelagic or open water preferring fish in blue. Green is the littoral or sort of plant associated species, and then benthic are bottom dwelling. And so, you know, that top row is showing you for cash and Lindsay, the overall catch. And in cash slough, you can see it's pretty variable, but it is declining over that decade. Lindsay slough just has low catches and it's pretty stable through time. And then below that top row is showing you the relative contribution of each habitat group. And you can see in the middle of our study years, that plant-associated fish group is definitely contributing more to overall catch. Okay, so now if we look at those sort of interannual trends through time and different variables, smoothed out to account for, you know, seasonal variation. The top row is showing you that submersed aquatic vegetation. You can see it increase. It does decrease at the end, and I will talk about that a little bit. You can see water clarity in the second row, kind of following that temporal trend in vegetation. And then below we have our three fish groups. And so pelagic fishes in cash slough, you can see are very wiggly. That's largely driven by threadfin shad, but there is a decrease in the pelagic fishes. Again, Lindsay slough, not much change there. The littoral fishes are both showing this kind of big increase towards the middle of that decade. And then the benthic fishes are, you know, I want to note the scale on those is different. They're a pretty small contribution to the fish catch, but they do show a little increase towards the end. And we did do this for individual species. I suspect I don't have time to talk about each of these species. So I'll just highlight that for the same species, they can be doing different things just in those different two slough systems. And even within the same habitat group, we saw a lot of variation in their trends through time. And I think it's more informative to look at how fish catches respond to the drivers themselves. So we can model catch as a function of vegetation density, which is what I've shown here. And I'll highlight a few things. So we have our sort of pelagic species and some of the native species decreasing as you increase vegetation. So I've highlighted threadfin shad, striped bass, and tule perch. And so those are all showing, you know, monotonic decreases as you increase your vegetation. And then no huge surprise, there's some non-native littoral plant-associated species that enjoy having higher plant density, and they've been increasing. So we see, you know, red ear sunfish, bluegill, and largemouth bass increasing. When we did this same exercise, though, for water clarity as a driver, there was a lot more variability in the responses, and it didn't map nicely onto kind of our prior expectations or maybe what we would expect based on other delta work. So we didn't see kind of really clear responses to water clarity with the fishes, at least. And I'll highlight, you know, these four littoral species. They're doing all kinds of different things in response to increasing water clarity. So there's a lot of complexity with that. And so I think, you know, maybe to wrap things up, I'll just say that, you know, with this decade-long study, we saw dramatic habitat shifts, and we saw fish species respond in kind of two general groups. So we have a whole set of species that really seem to prefer, you know, what I'm calling historic delta conditions, but I know that that's, you know, depends on your time window you're looking at. They prefer that higher turbidity, lower plant density, and that includes non-native and native species. And then there's a whole group of fishes that really do prefer those increasingly common delta conditions of high vegetation density and high water clarity. And so I'll kind of just reiterate the takeaways. We did really see a regime shift in these North Delta habitats. So we have droughts and plant expansion happening simultaneously. This shifted the system from low clarity, low vegetation to high clarity and high vegetation. And now we see that the remaining variability in clarity is driven by precipitation, so sediment loading within that system. And then the fishes were declining overall in catch, and we did see multiple shifts in the assemblage through that decade. And I think my final point will be that, even though we've seen a regime shift, the system still has, some dynamics. So I want to point out towards the end of our study in 2021, we had a mysterious die-off of some of that invasive vegetation. We have no idea what caused it, but it was pretty dramatic in Cache Slough. It happened in Lindsay as well. And in Cache Slough, it didn't come back for at least a few years. And what we see is that water clarity actually started to drop again. So partly this is due to having a really big wet year happening at the same time. But I just want to, put the point out there that it's a dynamic system. Our climate is going to continue to be dynamic. So habitat conditions here, it's not kind of a, all right, this is what it is now. We're going to keep changing. And with that, I'd like to thank our funders and, of course, my awesome collaborators at UC Davis, especially the Duran Lab, Watershed Sciences, were the ones, going out and collecting this data for over a decade. And with that, I'll take questions.
Question about the decaying composition showing. So in the case of Will, is it mostly the exception of atomic herbicides to versus all just non-native fish that make up that community?
The question is what's making up that littoral fish group? Yes, so I really was just talking about species that made up a meaningful contribution to the catch. There's more species that I didn't talk about, but you're correct. Yeah, it was almost entirely non-native species that are plant associated, sunfishes.
I do have a question. So given the fact that we may have more multi-omega droughts in the future, in spite of the whiplash you talked about, do you think this regime shift might be more like a tipping point and may change permanently, or it's too early to predict that?
Question is if I think the changes in the North Delta are permanent. Well, I don't want to make predictions like that. I mean, I'm certainly sure we're going to have another drought eventually that will probably enable more of these shifts to continue happening. But, I think the end of that time series did demonstrate that it can get shifted back if you can kill off the plants, I guess. But yeah, I don't know that the odds are good for that happening.
But do you have data where you've sorted out that the SED includes both native and invasive submerged aquatic plants? Have you sorted that out? Because I imagine historically we had native submerged aquatics out there. And prior to, historically prior to all this sediment that came in from hydraulic mining up in the foothills, the water clarity, I assume, was clearer than just a native community. So do you think that if you do you have data like that, or what can you say about that, the native versus invasive, or is it just the total mass that's the problem?
So the question, I guess, is about the composition of the submerged aquatic vegetation. So they were trying to distinguish different species contributing to that total volume. I don't think the volume metric I was actually using was exclusively non-native volume. So it's really kind of just the effect of having that vegetation there rather than the specific species. But it was almost entirely Brazilian waterweed in that SAB category.
Thank you for an interesting talk. I was wondering, did the native fish community bounce back after the SAB was taken out at the end of the study? Were the fish communities able to respond on that timescale?
So the question is if the fishes responded to that sort of late stage reversal in vegetation. There were certainly a few native species that did show rapid spikes. They're maybe not the types of native species we spend the most time thinking about, but like the prickly sculpin, hitch, I believe, there were a few native species that did rebound towards the end. So you remember that graph where the benthic fishes increased, they were the ones that showed that response. But again, that's not the fishes that people are often sort of managing for.
Excellent. So we are going to end our symposium group of talks with a bang, with a talk that has 3 different speakers. So really excited to invite up Emily Funk, Sarah Brown, and Aviva Fisk, who are going to talk about eDNA and eRNA, which has definitely come up several times today. Sarah is the unit manager for collaborative ecological studies at the California Department of Water Resources. Emily is the lab manager of the Genomic Variation Lab at the University of California, Davis, and Aviva is an environmental scientist at the California Department of Water Resources.
Hello, everyone. Welcome to the final talk of the day. You made it. So today, thank you for that intro, Rachel. I will be sharing the stage with my collaborators, Emily and Sarah, and we will be talking about using eDNA and eRNA as complementary tools for invasive species monitoring. So over the past several decades, extensive research on the use of environmental DNA, or eDNA, has shown huge promise for detecting rare species when traditional monitoring techniques can miss them. So eDNA or eRNA is genetic material that an organism releases from skin, mucus, feces, etc., into the environment. And when we say into the environment, we typically mean water, soil, or air. And just to remind folks the difference between DNA and RNA, DNA is double-stranded and it contains the genetic code for all living organisms. RNA is single-stranded and it translates DNA into proteins. And the single-stranded versus double-stranded distinction is important, and when we're talking about stability in the environment and how quickly DNA versus RNA degrade in the environment, and Emily will be talking about that more in her section. So both of these materials, eDNA and RNA, are very sensitive, so we can detect very low quantities of them contained in the sample. And in this intro, I'll be using eDNA as a blanket term for both eDNA and eRNA, and each of us will make the distinction of which one we're talking about in each of our sections of the presentation. So as I mentioned, the main types of eDNA samples are water, soil, and air. And within each of these types of samples, there are multiple methods of filtering and collecting the sample. Today, we will only be talking about water samples. So there are many different uses for eDNA for detecting rare species, monitoring biodiversity and climate change, pathogen monitoring, and what we'll be focusing on today, which is invasive species monitoring. So just a bit about how eDNA is collected and processed. We go out in the field and we take a water sample. Sometimes that's as simple as just taking a grab sample, so just filling a bottle with water and bringing it back to the lab. And sometimes we use specialized pumps and filters in the field to help us take these samples. So you can see an example of one of those pumps pictured on this slide. Once the sample's been collected, we bring it back to the lab for DNA extraction. Extraction is basically where we break the cells open so that the DNA is accessible for us to analyze. And then once it's been extracted, we can move forward with several different types of analyses. Some are for single species detection, so that would include qPCR and Sherlock that Emily will be talking about. We can also do multi-species detection, so that would be detecting the entire community of eDNA in a single sample, so representing the whole community there. And that's called eDNA metabarcoding, and Sarah will be talking about that. So some advantages of using eDNA and eRNA are that it's a non-invasive sampling tool, meaning that we don't actually have to capture or handle the organisms that we're looking for, so it's less stressful for them. It can also detect recent species presence. This is a question that has come up a lot in eDNA and eRNA research is, oh, well, what if you're detecting something that was there 10 years ago? And what we found in our research in the Delta is that these signals for eDNA and especially eRNA are really ephemeral, and they actually like dissipate pretty quickly. So when we detect things, we're sure that they were there recently. It's also very sensitive, as I previously mentioned, which means that there's higher probability for detection of secretive and rare species. And in the case of invasive species, we have the ability to detect them early on in invasions. As I also mentioned, we can do single or multi-species detections from a single sample. It is often less labor intensive than traditional monitoring, which means it can also be a lot more cost effective. And an additional benefit of eRNA is that it's only shed by living organisms. However, it comes with other limitations that Emily will talk about in her section. So some limitations of both eDNA and eRNA. As I mentioned, there's potential for degradation in the environment, which could be seen as a good thing if you're really wanting to look for recent species presence, but also we have to think about that in terms of being able to actually detect what we're looking for. Contamination risk is high in both the field and the lab. However, we have ways of dealing with this. One, we have procedures to make sure that contamination doesn't happen, but we also have controls that we use in the field and the lab. These are basically blank samples that we expect to not have anything in them. And if we see the DNA in them, then we know that we had some issue in our analysis, either in the field or in the lab where contamination took place. And then we can say, you know, this could be a false positive. And that's how we can really detect false positives and kind of keep them out of our reporting, especially for invasive species. So that is a thing that a lot of people bring up with eDNA, worrying about contamination, and it happens, but we can account for it and we can identify it. Also environmental factors, so turbidity and hydrodynamics. High turbidity can affect our ability to analyze a sample if we have issues related to the suspended sediment in the sample. And we also have to account for hydrodynamics of the system we're working in. So we want to make sure that we're actually capturing the sample, and we have to think about that a lot in the Delta in terms of flow and direction of water movement. We also cannot do any, we can't get any individual data from these samples yet. This is an area of active research, but as of right now, we can only do sort of community level analyses, presence, absence. We can't get population size, age, structure, reproductive status, all of that stuff, again, active area of research, but that's not something that we are currently capable of doing. So in terms of management of invasive species with eDNA and eRNA, our goal is to be able to detect invasions early. We've heard a lot of people talk about early detection and rapid response, and that's really the main driver of us doing this work. Can help us to direct eradication efforts and also to determine the efficacy of control and eradication efforts. So if we think something has gone from an area, we can go and continually take water samples from that area to make sure, like, is it showing up again? So we really want to use it as a complementary tool to fight against invasive species along with traditional monitoring techniques. So now I will get into the first of a couple of case studies that we're going to talk about using eDNA and eRNA to look at invasive species in the Delta. And I will be talking first about golden mussel eDNA. And since we already had presentations on golden mussels, I'm not really going to go into this too much. I think most of you know it was detected for the first time in October of 2024, and it's native to China. Some of the ecological impacts, again, I know we already kind of went through this in the previous session. So food web disruptions, changes in composition of benthic invertebrates, water chemistry changes, water quality impacts. I think most people here are familiar with all of the issues related to golden mussels. Also lots of economic impacts, mostly related to structural damage and impaired water conveyance capabilities. And I think you all have probably already seen this photo as well of water quality, probably several times today, of water quality instrumentation encrusted with golden mussels. And for just to go over the invasion timeline, basically golden mussels were invading Asia in the 1960s through 90s, and then they showed up in South America in the early 90s, and then the most recent invasion before North America was Brazil in 1999, and then they showed up in the Delta, Port of Stockton. So, and then for our response timeline, the first visual confirmation was done just a couple days after the first unknown mussels sample was collected at the Port of Stockton. And then just a couple days after that, we had the first genetic confirmation by UC Davis Genomic Variation Lab, as well as California Department of Food and Ag. And then DWR began monitoring using eDNA at the beginning of November of 2024. And since then, there's been lots of other agencies and groups that have continued to do eDNA monitoring work. And the main point that I want to make here is that we really were able to ramp up our efforts really quickly for this. So between October 17th, when it was first detected, and November 1st, by November 1st, DWR was ready to go and ready to start testing. So that was just a couple weeks that we were really able to kind of ramp things up and be able to help with monitoring the spread really quickly. And that's one of the advantages of eDNA. There's a lot of existing resources already for species of interest. And so we can kind of get things rolling really fast when there's like a new invasion. So early detection is key for managing aggressive invaders, as we all know, and genetic tools are really amazing because they can actually detect the arrival of just single veligers. So just a single larva floating around, we can detect that, which is pretty amazing. And I also want to highlight that there's been a lot of collaboration and communication across different agencies. So both government and academia. So the taxonomic ID was done by DWR and Smithsonian Institute. Genetic confirmation was done by UC Davis and CDFA. And now there's, as I mentioned, there's a lot of eDNA monitoring going on across many agencies. This is a really great example of collaboration across multiple levels of agencies and academia. Again, I think everyone has seen this map. This is the California Department of Fish and Wildlife map showing the spread of golden mussels. This map shows both visual and eDNA detections. And if anyone wants to see a map of just eDNA detections, we can provide you with that. Let us know if you want to see that, and we can do that. So before I hand it off to Emily to talk about Nutria, I just want to underline that one of the great things about eDNA is that we can really leverage existing monitoring efforts and just add genetic tools onto it. So we have a lot, specifically at DWR, we have lots of people going out regularly to monitor for water quality and doing other studies across the Delta. And we can basically just say like, hey, take this with you. Here's an SOP, go out, take this sample. And none of these SOPs are difficult, like anybody can do them, and just bring us the sample and we can analyze it. So it's a really great way for us to just kind of be able to ramp up really quickly and take these samples in a really low effort way. And it's really helped us to be able to track the spread efficiently. So with that, I will pass it on to Emily, and she's going to talk about Nutria.
Oh, that one, okay. All right, so I'm going to talk about Nutria and descendant mussels actually today, and kind of the comparison of developing some genetic monitoring methods for both. So yeah, so I'm going to talk about the development of Sherlock assays for the detection of invasive species. Okay. Oh, okay. I'm figuring this out. All right. Sherlock, it's a CRISPR-Cas method. It was originally developed as a point of care method for disease diagnostics. There's a COVID Sherlock test. It uses Cas13, which is unlike CRISPR-Cas9, which is the genome editing Cas. It does not edit a genome. Instead, the user designs a guide RNA specific to your target of interest, your species of interest. That guide RNA will find and bind to the target if it is in your sample. And then the Cas13 will start cleaving all single-stranded RNA in the reaction. So we include a fluorescent RNA reporter in our reaction. So it will be cleaved and give off a fluorescent signal, indicating the presence of your target species if it's there. If it's not there, no fluorescent signal. So the GVL, so the Genomic Variation Lab, and the GEM labs at DWR have designed and optimized Sherlock assays for use in ecological contexts, such as detecting and monitoring invasive species using eDNA and eRNA as sample input. And the reason why we use both eDNA and eRNA, Aviva alluded to this earlier, is that RNA will degrade faster in the environment, and it is only shed by living organisms. So it can help us improve the spatial and temporal resolution of eDNA detections. And an important thing to note about Sherlock is that it is rapid. All of our assays are completed in less than an hour. It's field deployable, so it is the reaction is takes place at one temperature, 37 degrees. So you don't need a thermocycler. There are a few different fluorescent readers that are portable you can take with you into the field, and it can be done by anyone. So a lab would prepare and provide a master mix, but then anyone can add their sample and run the Sherlock in the field. So our end goal is in-field detection of invasive species using eDNA and eRNA as sample input. And like I mentioned, right now we're working on 2 Sherlock assays for invasive species, one for descended mussels, quagga and zebra, and one for Nutria. And I'm going to talk about the progress and challenges we have had for both. So just a quick kind of overview. I've got a bunch of graphs to show you. The initial Sherlock assay design, we use tissue extracted DNA and tissue extracted RNA to design a species specific assay that is also sensitive. So for our descended mussels, we've got species on the x-axis, and you might not be able to read that, but the blue and the purple box are zebra and quagga mussels, and then the y-axis is relative fluorescence units, and you can see that we get fluorescent detection of our target species and not our off targets, which are shown in green, and then that little bar is just a control. Similar results for RNA. Again, we had very great results for our RNA, and so there's not a lot of variability, so our boxes are mostly just lines. But there are those 2 lines at the top for zebra and quagga. Unfortunately, we couldn't get off-target RNA because of the difficulty in obtaining tissues preserved properly for RNA extraction, but it is the same assay. So we assume that we will not get off-target RNA detection. And then we also do a sensitivity test using a synthetic RNA or DNA molecule. They don't make them for RNA actually. And we are able to detect down to a single molecular copy. So it's a very sensitive assay. And then for Nutria, similar success in designing the assay. So it's specific. Here, I apparently didn't include our controls, but that small pink box is our off targets and the yellow box is our Nutria. So we're specific for both DNA and RNA, and then again, sensitive down to a single molecular copy. So in theory, we should be able to detect low concentrations of DNA and RNA in an environmental sample. So next steps are to optimize the Sherlock assays with actual eDNA and eRNA samples. So for our descended Sherlock assay, the optimization was fairly straightforward. We didn't have to make any changes, and we received extracted eDNA samples from the Met Water District for quagga, and then from Stephen Spear with USGS for the zebra mussel water. And we got beautiful results. So this is actually a graph of our Sherlock assay happening over time. So time is on the x-axis. We run our Sherlock assays for an hour. And then again, fluorescence is on the y-axis, and we get, or we reach about maximum fluorescence at about 40 minutes. And then we had similar success with eRNA. There's a little bit more variability, which is not unsurprising given the low concentrations you get with environmental samples, but that's why we always run our samples in triplicate. However, Nutria, we're a different story. As I think everyone knows, Nutria live in swampy, turbid waters. And thus far, we have not had any luck with detecting our eDNA or eRNA. This picture here, that's literally like Nutria poo that we are filtering water right next to. So you'd assume that we definitely have eDNA in that sample. We've tried multiple different water collection methods and extraction methods, and we're still working on it. It's just swamps are hard for eDNA, eRNA. They probably doesn't preserve it very well. The filters get clogged really quickly, so you can't filter a lot of water in order to like capture that DNA or RNA. So you could get a lot of inhibitors, which are just preventing the Sherlock from running properly. So we're still working out the kinks for Nutria, but Dracennids were great. So there are challenges, right, to working with eRNA and eDNA. You can have a highly sensitive assay for detecting eRNA or eDNA, but you have to be able to collect it first, right? So environments that could present a challenge are highly turbid areas, which could lead to poor preservation of your DNA or your RNA. Water flow can have an effect. But then there are also systems like the Mussels that appear to be really good for implementing eDNA and eRNA monitoring. And our actual next steps or our next challenge is to develop an in-the-field eDNA, eRNA extraction method. And then ultimately that would allow us to bring this assay into the field and detect invasive species as quickly and sensitively as possible. And now I'm going to hand it off to Sarah.
Thanks, Emily. Okay, and I'll be talking about the Sentry program next. And so just to get everybody set up. So Sentry stands for Surveillance of eDNA for Notification of Target Invasives. And this is our new pilot program for environmental DNA monitoring of invasive species. And so Sentry spawned out of necessity. As you know, in October of 2024, golden mussels were detected in the port of Stockton. And this was devastating news, both in an ecosystem and an economic front as well. Invasive species threaten to alter ecosystems and damage aquatic infrastructure if they get established. And California natural resource agencies were caught off guard with the arrival of golden mussels, and we've been laser focused on other invasive species, but not necessarily golden mussels. And so in order to avoid other invasive species from taking hold in the delta and threatening the ecosystem and the infrastructure, we need to be proactive in early detection of invasive species. So early detection of invasive species means a greater chance in preventing their establishment and a greater chance of eradication, as you can see on the invasion curve that you've seen before. And traditional monitoring for species, including individual trapping and observations, they might miss rare or secretive species like early invaders. And so environmental DNA is a really sensitive tool, as you can see from Emily's and Aviva's talks earlier, that can provide a really early warning alert about new invasives. So some examples of eDNA in management of other invasives include tracking the leading edge
of the invasive silver and bighead carps in Wisconsin, and using airborne eDNA to detect invasive mosquitoes in Maui. So these efforts are aiding traditional methods in early detection. And so now we add DWR beginning this pilot program for eDNA monitoring of aquatic invasives in the Delta. So once again, I'll just state what Sentry means. So surveillance of eDNA for notification of target invasives. So 2026 will be the pilot year for this program. And our goal is to sensitively, rapidly, and efficiently identify new invasives from environmental DNA in the San Francisco Bay Estuary by leveraging existing monitoring efforts when feasible. So the scope of our pilot study is that we will be sampling quarterly throughout 2026 at high target sites. And because it's just a few of us that are working on this project, we have 4 sites that we will be starting with in this pilot year. And they were picked based on the proximity to ports or shipping lanes or the State Water Project. And so we have sites at the Clifton Court Forebay in pink, the Port of Stockton and Port of Sacramento in, what is that, green and orange, and then Chips Island in blue. And so we will be coordinating with groups at DWR, for example, the continuous and environmental and discrete environmental monitoring groups, along with others as they're going out and collecting their water quality samples. They will filter 3 replicate filter samples for us and then take them back to our lab. Because we are looking for new invasive species, we need a broad approach that will allow us to detect as many species as possible. And so we'll be using a meta-barcoding approach, which aims at identifying many different species in one sample of water. So we'll utilize barcode regions of the mitochondrial and chloroplast DNA, which are unique identifiers to specify taxonomic groups. And we focus on regions of DNA that are the same across individuals of a species, but they diverge in different species. And so just in a nutshell, we'll have DNA that will be extracted. We will amplify the DNA with from those regions of interest, those barcodes. We will sequence them, so amplify them up and sequence them. And then we will compare them to a database of other species that we know that we're looking for. And then we will identify the species that are present in that water sample. And so we have focused on 6 different taxonomic groups that should cover all potential invasives. And we've highlighted just a few of the invasives that we'll be looking closely for. And so the taxonomic groups are very broad: crustaceans, fish, mammals, reptiles, algae, jellyfish, mollusks, and plants. And our barcodes will be able to identify all species within these taxonomic groups, not just the invasive ones. And we'll end up essentially with a snapshot of the biodiversity for these taxonomic groups at each site. And so the next steps for Sentry will be obviously to collect our samples throughout the year and to process them. But then in thinking about moving forward, questioning whether or not we need additional sampling locations, if we can move beyond that 4 and increased frequency perhaps, and then potentially involving different eDNA collection methods, so testing passive samplers as well to our active filtering. And then adding additional relevant taxa if we find that there's, we might need to change some of the primers that we're using in order to reach a broader array of invasives. And then coordinating with other agencies for a plan if invasives are detected. And with that, I'd like to thank all of our collaborators on all 3 of our projects.
Thank you very much. My question, I work with a lot of state coordinators in regions outside of California. I was doing the same thing. And a lot of them are hesitant to use eDNA as like the sole thing to make sure there's some kind of management response or, you know, regulatory action declared something invested. I'm curious if you guys, with your experience, think that's the right approach, you know, to then wait till we have subsequent developer or develop detections if we're talking about mussels, or are we getting to the point of the technology that we should be more reliant on eDNA or RNA as just really detecting why you can trigger those management activities from them. And you have previous regardless.
Yeah. I don't know. Okay, so the question, I don't know if I'm the right one to answer this, but the question is whether we should be using eDNA or eRNA as the only method to trigger management action. I don't know. I think the methods are getting a lot better and a lot more reliable. But that being said, if you're trying to do kind of like a targeted, I guess it depends on your response. Like if you're talking about mussel aside, you have to have a place to apply it. And with eRNA, I guess the hope is that eventually the kind of the location of the sample would be a lot closer to where the actual animal is, but eRNA is a lot newer than eDNA. And I think eRNA has a lot, has more to go before it's as reliable as eDNA. That's what I got.
So yeah, that is a big, scary and loaded question, I think, but I will just highlight that, well, the methods are getting there and they are becoming a lot more reliable, as Emily said, but we actually have, for the first time, in the current ITP, there is a mandate that includes using eDNA as one of the monitoring methods. I won't go into that project in depth right now, but just to say that like it clearly is being more and more accepted, the fact that it's actually a mandated activity in an ITP now for the first time. Again, that's the first time in North America that that's happened. So that's really exciting and just kind of emblematic that with all the research and evidence that has piled up over the years, that it really is becoming more accepted as like a sole monitoring tool. But again, I think it depends also, on like the risk benefit of whatever the response is. You know, I don't want to say like, yeah, you detect it, go ahead and like, put a bunch of, mussel or whatever in the water. I don't really want to say that, but anything else?
And I just wanted to add that with the Century program, that is our thought, that like we will use the eDNA as a first detection method, and then we will go back out and do extensive surveys to look in the area, see if we can find actual individual observations of those taxa.
So my question is about the Century program, the plants you have to make those organisms. I'm wondering how you selected the plants that you did to do the testing for the Delta, because like water lettuce, Pistiosporidium stradioides, I don't even know of it occurring in the Delta. And where we have detected it, like I picked it up a couple times on the Russian River, it doesn't survive the winter. So I mean, we have, I just wonder, and then I'm not sure what marsh weed is, what species. I just wonder why those species and how that selection process went.
Yeah, so just to start off, sorry. Always forget. So the question is about the types of plants that were selected that were selected for the Century program. And so we looked at a list of potential invaders into North America, potential invaders into California, looked at the CDFW website of things that the CDFW is concerned about. And we have a list of plants that we are concerned about, but the primers that we're using will amplify all aquatic plants and even beyond that, I think. And so any plant DNA that's in our water sample, we will be able to amplify. So if it's not necessarily on the list, that doesn't mean that we won't pick it up. Yeah, which I just picked some random ones. I don't know plants very well. So I just put some random ones up there that